Thank you, Vice-Chancellor. Now I'd like to call upon the Dean of the Faculty of Science. I should add here that Professor Chetty has been instrumental in driving this process in many ways and got us to a point of making sure that the symposium did happen. In my regular meetings with him every 6 months, he would say, "Is the Ballinsky Symposium happening?" And I would have to say, well, we postponed it again. But I think now we have the opportunity to do justice to that. Thanks, Professor Chetty, not only for your support in driving this, but also helping to raise the funds to which we can actually have the celebration today. Professor Chetty.
Is that connected? Yes, thank you. Thank you very much, Professor Pillay. Yes, indeed, this has been a long time in the planning. Vice-Chancellor, Professor Zeblon Bilakazi, and Her Excellency Madam Ambassador Abravitova, it's really delightful for me to be addressing you today. It's been 2 or 3 years in the planning, and this was originally meant to be a part of our centenary celebrations. So very glad that this is happening, and thanks for your tremendous efforts to the School of Animal, Plant and Environmental Sciences headed by Professor Neville Pillay. Ambassador, thank you for your resilience and for your strength. Scientific cooperation is so very necessary for us to build bridges across many different barriers and difficulties, conflicts in this world. And I certainly long for the time when peace can descend on your great country. Ukraine has a great tradition of excellence in science. I myself, and so with the Vice-Chancellor, am a physicist, and we certainly recognize the tremendous contributions that your great country has made to the field of physics. I thought I'd put that in first before I talk about the biological interests that we have before us. Scientific cooperation is essential, and I hope that we will continue with cooperation through these difficult times, because this is going to be very, very important for the rebuilding exercise which I hope can begin much sooner rather than later. I'm delighted today to recognize Professor Emeritus Barry Fabian, and Barry was mentored, of course, by Professor Boris Ballinsky. And this is what the university is all about. It's building on the next generation, because we know that the current Deputy Vice-Chancellor for Research and Innovation, Professor Lynn Morris, who unfortunately could not be here today, was mentored by Barry himself, and so are many of the other academics, Marcus Byrne, together with Neville Pillay, also mentored by Barry Fabian. And we will hear a little later from Professor Hazel Sev, who's a honorary professor now from the university, but a graduate from Wits, an alumnus, currently Dean of Science at Northeastern University, and she also pays a lot of homage to Barry Fabian. So this is really what we're trying to achieve, is to build on the next generation of science. I also want to recognize Mrs. Helen David. I was really delighted to meet with you, and thank you for being here, really, to make the connection with Professor Ballinsky, a very real one for us this morning. So colleagues, and I do want to recognize those who are joining in online from Ukraine and from elsewhere in the world. We are gathered here today then to celebrate one of our best known and respected embryologists of the 20th century, and that's a worldwide recognition undoubtedly. He laid the foundations of developmental biology as we know today, has Boris Ballinsky. He completed his undergraduate studies at the then University of St. Vladimir, subsequently now the University of Kiev, about 100 years ago, a little more than that, 1923, would you believe? And as an undergraduate student at the age of 19, he did some incredible things. and this is where you see the curiosity of the young Ballinsky coming through. He transplanted ear vesicles into the lateral body wall of newt embryos and discovered that a supernumerary limb developed. So that must have been a startling discovery, but it shows the inquiring mind at a very young age. And he published his first scientific paper in 1925 as an undergraduate student. Quite a rare achievement, really, and probably Kazi will remember Richard Feynman. He was of that class as an undergraduate student. The Nobel laureate Richard Feynman, also quite prolific as an undergraduate student, and we need to pay attention to this. We need to inspire students as early as possible and create the opportunities for success. And here we see Ballinsky already at a very early age being inspired. So he continued with this work with great passion throughout his scientific career. So the very genesis of this was so many years before. And he obtained his doctorate in biological sciences from the Ukrainian Academy of Sciences in 1935. But it was at WITS that his embryological teaching and research gained traction. In his textbook in particular, and you will see a copy of that in the glass case in the foyer, entitled Introduction to Embryology, which was first published in 1960, before I was born, it turns out, secured his international reputation, undoubtedly. The book was sculpted from his courses that he taught here at Wits University. The book was published in 5 English editions, 1960 through to 1981, and it's translated into Japanese editions, 2 Italian editions, and a Spanish edition. The book had a global impact that became the foundational textbook for embryologists around the world for many years, and no doubt influenced the education of countless number of students around the world. He also became a keen entomologist, it turns out, collecting butterflies, stoneflies, and dragonflies, and you'll see some of those dragonfly collections and I'm quite amazed that they're so robust that they remain fairly intact. You've got to tell me how that happens, Neville, because I'm just a theoretical physicists, don't quite understand these complexities, but really fascinating for me to see these dragonflies that Ballinsky collected being preserved so very well. He was also a pioneer, as we heard from the Vice Chancellor, in the applications of electron microscopy. So in this way, he was ahead of his time. I have no doubt about this. In 1956, it turns out that Wits University acquired an electron microscope. We still have that facility today, which is a multidisciplinary facility that serves many different disciplines. But I can imagine way back then, it would have been quite intimidating for a biological scientist to enter into an electron microscopy lab and start doing things there. The technology might have been quite different from what one would be accustomed to. It enabled him to advance embryological studies, which he expanded to include several other animal taxa. He became probably the first person to practice biological electron microscopy in South Africa. And famously, he was a founding member of the Southern African Electron Microscopy Society and became its president as well. So I think that's quite remarkable. So upon retiring in 1973, Ballinsky continued his studies on the variation of wing-colored patterns in butterflies, demonstrating his keen observational abilities in adherence to sound evolutionary biological principles. He published 5 papers in 1972 to 86 on this topic, on color variation of butterflies, conducting breeding experiments, would you believe, in his home here in Johannesburg, really showing his dedication. He donated his collection of several thousand specimens and his extensive collection of reprints as well to the then Transvaal Museum. He was head of biology in 1954, and that's a seat that Professor Neville Pillay holds. He was also Dean of Science, more or less at the year that I was born, it turns out. So we really look at a colleague who's left a lasting impression here, 65 through to 67. So colleagues, we are very proud then to host the Ballinsky Symposium, a Ukrainian-born South African. He belongs to both of us. We'd like to have more of him than you, but for today we're going to share him with both countries, both great countries and great friends. He embodies the spirit of being a Vitsi. And a Vitsi is something very special, I can tell you. Once a Vitsi, always a Vitsi. We have a certain passion about excellence. And he certainly embodies that and set the foundations, I have no doubt, for excellence, not only in the zoological field, but more broadly within science and the university. The significant impact of experimental biology at WITS led to his global recognition as an embryologist. His research spans several taxa, from the anurans, that's frogs, to insects, sea urchins, and so on. And of course, South Africa would have been just a fantastic real live laboratory for him. Of that I have no doubt, in his adopted country. The breadth of his zoological research is astounding and reveals his focus and agility. So in the next 2 days, colleagues, we will honor Professor Ballinsky's legacy. We will hear presentations broadly in developmental biology, evolutionary biology, and ecology. I wish you all a successful symposium. Thank you.
Thank you, Dean Chetty. I'm just going to check with Rochelle. I can see you at the moment, Rochelle. We're slightly ahead of time. I don't know how we're timing it with the people online. Can we continue with the program? I think those words by the Dean summed up Professor Ballinsky in his widest reach within the university and globally. So now it's my privilege to welcome Excellency to speak on behalf of Ballinsky in relationship with the Ukrainian people. Welcome, Your Excellency.
Good morning, ladies and gentlemen, Vice Chancellor and Principal of the University of Leeds, friend, Professor Chetty, friend, friends here, dear academics in South Africa, in world, but of course in Ukraine. We are finally at this moment when we are celebrating the legacy of Ukraine South African scientists. I want to say special welcome to a very special guest today, Miss Helen. I think she is rather host than guest. And I want to express our gratitude for the example of how one has to carry the history, legacy, and honor of such a father that you have. We admire you. You gave us a lot of impetus not to give up and to have this important scientific event. Thank you so much for carrying so carefully. And applause, please. I also want to express huge gratitude to the Ukrainian Association of South Africa, because the dedication that Ukrainians, the very tiny but very strong Ukrainian community in South Africa, preserving every piece of our legacy everywhere in the world, particularly in South Africa, contributed to this event is very significant, and I want to appreciate it as well. Thank you. So when you are coming to universities, to any scientific event, usually you see very boring faces, sorry, ladies and gentlemen, but I see and feel the vibe of today's event, that really we came here to celebrate something, and it definitely gonna be very inspiring one. Or maybe I should attend such events more often. And that's something that we will conclude with today. My assistants wrote for me long speeches usually, I was told by Professor Vilakazi that we are not here talking a lot. We are talking straight to the point. So I will also try to be very straight to the point. Indeed, we were planning this event for a long time, and different interventions came across. First one was pandemics, then the invasion of Ukraine by Russia and the continued war, which, let me remind you, didn't stop yet. You know, maybe everything that is happening in time. And probably this is the very timely that we have this celebration today in 2024. We're still celebrating the centenary of WITS, and we still congratulating one of the most respected universities of the world. It's our honor to be on this platform. But we also have to remind to ourselves that this time when Ukrainian scientists in Ukraine are pursuing the very same goals, keeping up with the challenges of our contemporary world, under the massive attacks of Russians, we continue to be dedicated to the legacy of those scientists of Ukraine that made the huge input to be known, but also to those who today can make difference, who can change this world for the better. And I want us to remember that as we are talking today, our scientists, our universities are very dedicated to continue to make their input, and we're grateful that during all these difficult times they were receiving a lot of support and platforms, particularly through the WITS University. Let me say that being the brilliant Ukrainian African scientist, biologist, embryologist, entomologist, author of more than 140 scientific works, what Ukraine is really caring and very proud of is that at that time 22 of them were written in Ukrainian language. The alma mater of Mr. Ballinsky is the University of St. Vladimir, which is today known in Ukraine and in the world as the Taras Shevchenko University. This was the time when Mr. Ballinsky was the first one to teach zoology in Ukrainian language at that time. For us today, not only in South Africa, in Ukraine, it is very important to research the memoirs of this scientist, because these are the very rare memoirs and works when between the two world wars, the brilliant brain of Ukraine was able on his own eyes carry, first of all, to carry the self-identity as Ukrainian, but also to, on his own eyes, to see the suppression by Soviet regime of Ukrainian identity. He was the victim of that regime, that one who had to flee from his own country. And that's exactly how he, at the end of the day, found the home and, if I can say so, cradle of his scientific work in this beautiful country. Absolutely, we are sharing a lot of similarities, Ukraine and South Africa, and it already was said that Boris Ivanovich was a big admirer of nature, and it would be very difficult after Ukraine to find another country with another beautiful nature which could encourage him to make those researches that he was fond of. And South Africa definitely was the right choice. And probably we, as Ukrainians, have to be grateful for that hospitality that South Africa granted to him and gave opportunity to open all his talents to the world. I want to also remind us that Boris Ivanovich was the person very open to the new technologies. And every generation is facing the challenges of new technologies and perceiving it differently. Today we are facing the new IT wave of researchers and Ukrainians are very open to it. Yes, we're in the circumstances where we have to move forward in order to survive, because if we don't use the new technologies, then we are at risk of being eliminated. But at the times of Mr. Ballinsky, the new technologies also were a challenge. And I noticed in the vestibule of the university that very famous microscope that Mr. Ballinsky was working with, he made a revolution. And I think that this is a very strong Ukrainian legacy and heritage that we are showing today. And he is the bright example of that. Using IT technologies, using the new technologies, using the not being scared of new approaches in science. This is something that Mr. Ballinsky showed to everyone. And this is something that we are going to research even more. You know, I want to, being here in Wits University, I would want to emphasize that we did discuss with professors the huge potential in cooperation in different sciences, including physics, between Ukraine and South Africa, and we are very dedicated, and we understand that this is the way forward for future of Ukraine, but also of South Africa. I hope that after all not boring and encouraging interventions for this couple of days that the symposium is happening, we will see dedication from other scientists to build firm bridges between our universities. The Embassy of Ukraine in South Africa is here for this purpose, and we are very eager to build these bridges. We have a tradition of learning and teaching on both sides of our students. For your information, before invasion by Russia of Ukraine started, we had around 25,000 students from abroad, particularly from the African continent. And that gives you an idea of how many scientists Ukraine helped to raise on the continent, and I really want us to continue this approach and this work already now. We don't have to wait when Ukraine wins the war. We have to prepare ourselves to the collaborations for the future of our country. Dear guests, I'm sure that this symposium, this long-awaited event, will be only the beginning of our two countries' cooperation. It will bring and build the fundament of which Mr. Ballinsky, Boris Ivanovich, put a big, firm, solid brick into this Ukraine-South African scientific relations. Appreciate your interest to this symposium, and hope we will see a lot of future collaborations all together. Thank you so much.
Thank you, Ambassador. Those were very inspiring words. What struck me was how Professor Ballinsky pushed boundaries, and I think that's what BITS is proud to do. And we hope that our students can do this into the future as well. It's now my great privilege to call up Professor Emeritus Barry Fabian, who will talk about his relationship with Professor Ballinsky over several decades, many years ago. Thank you.
It's a great honor to pay tribute to my teacher and mentor, Professor Boris Ballinsky. He was an outstanding broad-based biologist, a gifted experimentalist, and author of the leading textbook. Ballinsky was also a talented pianist who had studied at the Kiev Conservatory of Music. In honoring Ballinsky, one must pay homage to his alma mater in Kiev and his luminary mentor, Professor Smalhausen. Another star to be in the Kiev days was Dobzhansky, who would integrate Mendelian genetics and evolutionary theory. It was the golden era of Drosophila flight genetics. Ballinsky writes that Dobzhansky, his senior by 5 years, knew him quite well. While embryonic development was Ballinsky's early defining thrust, he had a parallel interest in entomology, which began with his butterfly collection as a young boy in Kiev. In those years, he was also enthralled by Brehm's Life of Animals of the 1860s, describing travels and observations of animals in Africa. That he would one day be venturing into the African wilderness was the stuff of dreams, but it was to be. He arrived in South Africa in 1949, and nature beckoned. He had only to stroll from his house into the nearby open land, where rain pools filled up during the wet summers and hosted breeding bullfrogs. Together with his son John, a student at WITS, they spent time observing the frogs, and a joint publication followed. John Ballinsky went on to head the Department of Biochemistry at WITS. In 1960, Ballinsky ventured north with our honours class and staff. The main aim was to examine the effects of the rising waters of the newly built Kariba Dam. My fellow student, the late John Munro, wrote in his autobiography that we went from Johannesburg through southern Rhodesia, now Zimbabwe, to the Zambezi River. We travelled in Land Rovers across almost trackless terrain. The area was uninhabited for hundreds of kilometres, and the wildlife responded accordingly. One could walk up to an antelope, which regarded one with interest, but were unafraid. And there were elephants and lions around. I too vividly remember that trip. On one occasion, Professor Ballinsky was out in front with a group observing elephant. When those ahead went into a sudden retreat, we joined the rout. An agitated elephant had charged. The Kariba venture was followed by a visit to the Marini Nature Reserve in Botswana. The reserve straddles the Okavango Delta and hosts a great diversity of plant and animal life. A special event each year in the zoology department's teaching and research was a visit to the Marine Biological Station in Yako Island off the coast of Mozambique. The trips would have reminded Ballinsky of his 1927 visit to the Merman Biological Station along the Barents Sea, where Schmalhausen sent him to study the rhythm of cleavage in a sea squirt egg, and of later visits to the Karadag Marine Station in Crimea. On one Inyaka trip, Ballinsky collected material for an electron microscope investigation of the mechanisms of cell adhesion in a developing sea urchin egg. He was a pioneer of biological electron microscopy in South Africa. As a student in Schmalhausen's laboratory in the 1920s, Ballinsky made his seminal discovery. He had grafted the ear rudiment of a newt into its fleck to study ear formation. To his amazement, a bulge with minute fingers extended from the implant. An additional limb was forming. If the implant was forward, it was a forelimb, and if to the rear, a hind limb. He had tapped into the head-to-tail positioning system in vertebrate embryos. 70 years later, a protein growth factor was found that induced limb formation, just like Ballinsky's graft. When I told a 90-year-old Ballinsky about the wondrous molecular finding, he replied with a twinkle, "I see I was most wise to have taken up butterfly breeding in my later years." The butterfly breeding project was no mere pastime, but a deep experimental study on the genetics of variation in butterflies. In anticipation of his retirement in the 70s, Ballinsky prepared the way for his new line of research on the South African garden butterfly. Where's page 6? Its caterpillars feed on leaves of the Kiolaria tree, which he was able to grow in his garden. He built a butterfly breeding shed, and his study became a research laboratory. Starting with single pairs, Ballinsky was able to rear a cria through 10 generations under conditions of inbreeding. In order to increase the amount of heritable variability, he irradiated the pupa with X-rays. He summarized his findings in a letter in February 77. What I think is really the main general result of the work I've done so far is the loose connection in my butterflies between genotype and phenotype. The genotype expressing itself in a variable way, which of course is nothing new, known in Drosophila. But the Drosophila is only one species, and I'm not sure it can be considered as a typical case of a wild animal. Ballinsky's crystal clear lecture course to our third year class came with 2 typewritten booklets entitled An Introduction to Biology. The booklets were submitted for publication to the W.B. Saunders Company. The first edition of a leading textbook was launched. I cherish the once-a-week visits to his home when he asked me to assist with the 5th and final edition. It would feature a concluding chapter on a theory of development recapitulation. By then, Ballinsky had started a new project on the classification of moths, which produced 5 papers. In January 1996, we honored Ballinsky's outstanding achievements at the first African International Developmental Cell and Molecular Biology Conference held here at WITS. The keynote address was given by WITS alumnus Sidney Brenner. Ballinsky at 90 was able to make a brief appearance. Boris Ballinsky gave several introductory lectures to our first year zoology class in 1954. We knew little about his earlier years, of his remarkable achievements and indomitable spirit through tempestuous times. That indomitable spirit is with us today. Thank you.
Thank you, Barry. That takes us way back in time. I'm reminded that Barry Fabian had a birthday recently. Barry, you might want to confirm it, but I think you are also close to 90, if I'm not mistaken. So, Barry, you might be 88, I think. Yeah. Yeah. You had a birthday last week, or was it the week before, Barry? Yeah. So I think we should give Barry a round of applause at 88. You certainly raised some of the big names in biology. Certainly Dobzhansky is someone that I read many years ago and was very intrigued by. And I realized that he had a strong Ukrainian background as well, shaping some of the neo-Darwinian theory in evolutionary biology. So it goes back. And because the legacy extends way back into the past. We're running slightly ahead of time. We've now done the informal part of the presentations. We're waiting for the first scientific presentation from Professor Hazel Sieve, who's in Boston, and she's decided to present live. So we just have to make sure that her timing and our timing works together. I don't see Rochelle. Maybe she's outside again, just to see if Professor Sieve is available. Is she available, Rochelle? Not yet. Okay, so we will wait a bit and see if she's able to connect. The time was meant to be 10:15, so if you want to stretch your legs and get another cup of coffee, we'll wait for you as you do that. But I'd like to, in the meantime, thank all our speakers this morning for setting the tone of the symposium. It's a very important part of what we were trying to do for many years. I've held many discussions with some familiar faces in the audience trying to set up the symposium. My initial discussions were online, and I remember seeing a lot of people from Ukraine online, and nice to see them here again. And as I said, it was a rocky road to this part, but I'm so glad that we eventually got to do this. So thanks to the Vice-Chancellor and the Dean, who I believe have to leave us, but the spirit of the symposium continues. As we wait for Hazel, Professor Hazel Sieve, you're welcome to refresh yourselves outside. And Zvinka, a colleague that I know, who's visited me a few times in Edwards, has made special cookies from Ukraine. So please try those cookies. They're out front, the colorful ones. Okay. If you don't have the program, the next speech is at 10:15. So we're talking about 20 minutes. Welcome back, everyone. That was the tea break, just out of interest. So we'll motor on with the rest of this program. And just confirming with Richelle at the back that we're all set to go. It's now a great privilege to introduce Professor Hazel Sieve, who was actually also from WITS, an association many years ago. And Hazel is joining us live from Boston. So it's probably early in the morning for her to do a presentation. The initial thing on your program said it will be pre-recorded. Late yesterday afternoon, Professor Sieve said she would like to do it live. So that's a great privilege. Just to fill you in some of the background, Professor Sieve received an honorary doctorate, I think, last year from the university. So there's a longer connection with us. So I'm going to hand over to the technical people and to then broadcast Hazel into the venue.
Anyone hear me? I see a nod. Do you see my slide? Okay, I'll start. One day, Barry, if you're in the audience, Barry Fabian, I apparently missed your talk. I'm really disappointed. I got up really early so I could hear, but I guess the time of your talk was changed, so I'm very sorry about you. So good morning, everyone. I'm Hazel Sieve, and I'm honored to be asked to speak today. The title of my talk is the extreme domain controls mouth, brain, and face formation. And you know who I am. I was born in Joburg. I got my BSc from WITS in chemistry and zoology, and my BSc art got a PhD. I was on the faculty at MIT for 2020, right at the Northeastern University, to become Dean of Science there. The research that we do focuses on craniofacial. And, you know, as was indicated, I'm a proud WITS alum. I serve on the board of the WITS Fund, and in the end of 2022, I was honored with a Doctor of Engineering honoris causa. So very honored to receive that from Vice Chancellor Zebulon Velakatsi, who I'm not sure if he's in the audience. I can't actually see who's there right now, but it's a real, very close to my heart to be a WITS alum, and a great honor to speak today. So you might wonder why I was invited and what's my connection with Dr. Ballinsky. Well, my connection is really through Barry Fabian, who was my most important teacher and mentor at WITS. And as I presume he told you, he was mentored by Dr. Ballinsky. So I'm Dr. Ballinsky's grandchild, and that's why I'm here today. Through Barry Fabian, I was introduced to the frog Xanopus, South African claw-toed frog that we kept in the zoology department at WITS, and that's where I learned about frogs and how to use frogs as a model to study vertebrate development. And my life's research has used this model. And over time, from the beginnings at WITS, I've become a world's expert in Xanopus care, in Xanopus techniques. And in that regard, I initiated the early development and cell biology of Xenopus course at Cold Spring Harbor Laboratories a long time ago. It's been running continuously with various leadership for 33 years. And this, we published a lab manual that came from this. The most recent lab manual, which totally changed from anything previous, was published last year with more than 1,000 pages. It was a huge project with hundreds of authors and hundreds of protocols. And I was editor-in-chief. I can't see the audience, so I hope that, Barry, you were there. I want to thank you in front of this group of people, illustrious people, that for being my dear friend and my guide all the way from Wits all the way to today. Thank you very much for everything you've done for me. And I realize that much of this stemmed from, oh, look, I see the audience. That's nice. Look. It was nice for a moment. I saw people clapping, Barry. Absolutely. Thank you. So look, can we keep it on? And there I see Zeblon. Hi, Zeb. So nice to see you. Thank you, Barry. I can't see you, but thank you for everything. It's wonderful to know you. There he is. Hey, Bear. I'm waving also. Such a funny angle. Okay. Well, better than pre-recorded. So here I am. Good. And here for you all to see is the cover of the Xenopus manual. It's the largest single organism project that Cold Spring Harbor Labs has ever done. And so we're very proud of it as a community, and I'm very proud to be its editor. And in the preamble, I talk about how I was introduced to frogs at Wits University and how they've been with me on my life's journey ever since. Okay. So let me move now into the science aspect. I would say we use development to study many things, but really we use the power of development to understand how the embryo is built, how organs in the embryo are built, and when things go wrong, how disorders arise. And in various studies using both frogs and the zebrafish model, we have been able to define some pioneer new treatments. I have been described as the mother of fields. I thought that was good. It's a little change from father of fields. I've been described as the mother of fields. And over the course of my career, I've started a number of different fields, some of which is one around a region of the embryo called the extreme anterior domain. That is very important in setting up the body plan, and particularly the face and the brain. And these are the ones, if you look, I have a Wikipedia page, and I gave a TED talk, and if you are interested, please do take a look and you'll learn about some of these others. But this story today has to do with building the head. And it has implications, as I'll tell you, for understanding microcephaly, which is a devastating birth disorder. The thing is, that the head is really an entity. We think about our own heads, and in ourselves, in these different animals here, there really is a kind of a dividing line between the head and the rest of the body. And during development of the embryo, you can really trace how the embryonic head is being formed. And it's really under its own set of controls relative to the rest of the body. The animal that we've used for these studies is the embryonic tadpole, or the embryonic frog, the larval frog. So here's a head-on view of a embryo, a larva at about the time we use them. Like ourselves, Xenopus have two eyes, a mouth, which will be important, and then this thing called the cement gland below, which is a kind of a marker of the front of the embryo. But a lot of the time that we'll talk today, I'll be showing you some head-on views of the frog embryo, which we've used to model human development as well as those of other animals. And what I'll do is to tell you three things. I'm going to talk about the extreme anterior domain. I'll tell you how the extreme anterior domain forms.
In progress.
And you'll see why it's interesting in a moment. But it's interesting, actually, because it's present in almost all multicellular animals. You can trace things all the way back to the sea anemone, to Nematostella, for example. Here's something a bit more complex, the acorn worm, the sea urchin, here's frog, and here's human. And you can find in each of these animals, you can find a region that is the extreme anterior domain. And in frogs and in all of these organisms, where the extreme anterior domain is present is where the mouth is going to break through. And so in people, this region here is exactly where your mouth broke through when you were an embryo. You can find this extreme anterior domain throughout development. You can find it starting from about the so-called neuralis stage, and it goes, here it is, it's boxed in red, it changes shape, and then it eventually in the tadpole stage, the purple cells form the anterior pituitary, the nostrils, and the mouth. And so you can see that this extreme anterior domain is present as the face is built by these green cells here, which are called neural crest. So that's an introduction to what this region is. It's this mesoderm-free region at the front of the embryo, present in pretty much all multicellular animals, particularly if you're, like phylogeny, particularly the deuterostomes. Now I'm going to tell you what this extreme anterior domain does. It forms the mouth. And in fact, when we started looking at this project, the mouth has been studied for quite some time. There are entries in human anatomy textbooks, but exactly how the mouth forms hadn't been looked at for really quite some decades. And so we reopened this field. We took this region that we had named and we reopened the field of mouth formation. And as I was doing it, appropriate for this symposium, really the only textbook that had any description of mouth formation was Ballinsky's book. And Barry, I used the edition of Ballinsky's book that you co-wrote with him, which I still have now, not right next to me, but I have that book, and it had actually quite a nice description of what was known about mouth formation, which was not very much, but it was there and it was very useful as we reopened this question of how your mouth forms. And of course, if your mouth doesn't form, it's a problem. So there was sort of both interest and medical reasons to be interested in this question. And over a lot of work, put some of the references on each of the slides, over a lot of work, we found some really fascinating things about mouth formation. And I'll go through this one summary figure with you to give you a sense of. These are, again, head-on views of the developing frog embryo. And so we're looking straight head-on as you're looking at me. We're looking head-on at the embryo. Here's its eyes, its brain, its and the blue cells here, blown up in these bottom panels, are going to form the mouth. What we showed was that there's a mass of cells, all sort of stuck together in this quite wide box initially. The cells then somehow know to sort themselves out and form what we called a pre-mouth array, two straight lines of cells that get a gap between them after a while, and then they kind of unzipper. And they unzipper in different places to open up to form an indentation that's called the stomodium, and that term is used in all animals, in all deuterostomes, including people. And eventually this indentation, the stomodium, breaks through, and that gives you the opening of the mouth. We identified many genes that were involved in controlling mouth formation and many signaling processes, but I'm not going to tell you about those today. What I will show you is a kind of cool movie that we took. So I'm going to show you on the left are going to be some confocal sections, microscopy sections looking straight on at the developing mouth, and that will be right here. And then we claymated the process so that you can see using some modeling clay that how it all works. So I'm going to start the two animations, and you'll see there's serial sections of the developing mouth. There it's elongated, and here in the clay we've got the blue cells elongating and then making little holes, opening up and opening up by the claymation, and there you go. There is the mouth that is opened from the clay and from the embryo itself. So that was really fascinating. And as I say, there are a bunch of genes involved, and we identified a number of interesting and important processes during mouth formation. But let us move on from that acknowledgement of that particular project and the relationship to Professor Ballinsky and Professor Fabian's excellent textbook to what I'll talk about for the remainder of the talk that came from an unexpected finding and turns out to be very important in how our faces are put together. And the topic is the control of face and brain formation by the extreme anterior domain. So I won't give you the discovery, but it was really quite striking, where we identified that this region, the extreme anterior domain in the purple in the red box, is not only going to form the mouth, it also is a signaling center, a group of cells that sends out chemical signals. Possibly also there is physical signals involved, but we'll just talk chemical signals, and also receives signals from the surrounding area. And this signaling process is really pivotal, is required for formation of the entire face of the embryo. Through subtractive cloning, we identified many signaling genes through which the extreme anterior domain acts as a major craniofacial signaling center. And the idea is depicted again here. Here's the extreme anterior domain at the very front of the embryo, sending out signals. And we envision that those signals form a kind of a sphere that goes all the way through the developing head up indeed into the developing range. And so there's a hypothesis that the extreme anterior domain has some range, some distance over which it signals. And what I want to do for the remainder of our time together is tell you how the extreme anterior domain controls both facial skeleton and brain size through a particular signaling pathway, which is called beta-catenin Wnt signaling, and that is controlled by another gene called Frisbee, which is an inhibitor of this beta-catenin Wnt signaling. So there's a signal and there's an inhibitor, and the story I'll tell you about involves both of those, but you don't have to appreciate that much. Just take a look at what I'm going to show you. So what I'll show you is in the regular embryo, there's the signaling center, the extreme anterior domain, and the outcome of its work, as well as many other cells, is a formation of the head of the embryo with its normal cartilages shown in blue and its brain shown in red. And when this extreme anterior domain is perturbed in some way, removed, or the genes that it is expressing are changed, you get a little head, get a small brain, and you get anomalous cartilages. And that's what I'm going to tell you about in the next slides. The first kind of assay that we used, and this work was done primarily by Justin Chen, a graduate student in the lab, was to extirpate, to cut out the extreme anterior domain. Xenopus embryos are tiny, but they're actually large enough that under the microscope you can do micromanipulation with various tungsten and glass needles. And so you can go in and cut out this region. It's quite small. It's about 500 cells, but you can cut it out quite precisely. The embryos heal very quickly. And then we looked at their faces, and we looked, as I'll show you in a moment, at the cartilages and their brain. And the kind of data you see is on the bottom here, where here is a control, and here's your regular tadpole little face there with its mouth and its proper width. When you cut out the extreme anterior domain, you see that the face is much smaller, or the head is much smaller. There's no mouth, and it's thin. You look from the top, it's kind of thin, and it's got a funny shape from the side. So when you quantify this, you can see that the width of the face really is much substantially lowered after you remove this small group of cells from the developing embryo. And that was one of the things that told us that this region was doing something, not just in itself, but to affect the whole face in its role as a global signaling center. We also looked at the cartilages and the brain. You can see here in the control animal, here are the cartilages stained with Alcyon blue, and they have there are certain types of cartilage and certain organization. If you remove the extreme anterior domain, this front cartilage it's there, but it's shaped all in a funny way, as well as the other cartilages as well. So there are decreases in the sizes of the cartilages, which are quantified here in these graphs, when you remove the extreme anterior domain. And when we looked at the brain, we could see similarly, especially for the forebrain, the telencephalon, there was a big decrease in size, also for the midbrain, and to a lesser extent, the hindbrain. And so these data told us that the extreme anterior domain was not only required for mouth formation, it was required for formation of the correct width of the head, for the correct cartilages and their organization, and for the various brain regions. And this was really the first demonstration that this region of the embryo was controlling a very broad set of structures that make up what we call the head, the face, and the brain. So we wondered what kind of signaling was going on, how was the extreme anterior domain telling the face to actually form? And Justin decided to look at a signaling molecule called Frisbee, and these are pictures of where the Frisbee gene is active in the embryo. If you're work in such things, these are in situ hybridizations with a fluorescent probe, but they tell you really where this molecule Frisbee is present. And the thing that struck us about it is that it's present really only at the stages we're looking at in the extreme anterior domain. So here are head-on views of the embryo, and you see this little pink patch is exactly the extreme anterior domain, and here's some side views. So this caught Justin's attention, and we started to look and see whether or not Frisbee was active as the extreme anterior domain did its work. And we hypothesized, just grab a little drink here. We hypothesized that Frisbee from the extreme anterior domain was acting as a long-range facial regulator, so a craniofacial regulator. So here we hypothesized this would be where the Frisbee protein was made, and we know that it's a secreted protein, that it leaves cells and it can move out from the cells that make it, and we hypothesized that Frisbee was acting over this whole region of the head to control face and brain formation. We tested that hypothesis using a kind of crazy technique that we developed in my lab that we call facial transplants. And in this, we take a donor embryo that has had something done to it. It's shown green here. It can be labeled green. It can have various genes that have been inactivated or overactivated, but there's something different about the donor sometimes, it depends on the experiment, from the recipient, which is unlabeled. And we can go in, really it's quite extraordinary, we can go in, cut out this little extreme anterior domain region and place it from the donor embryo, the green one, into a recipient embryo that's had its extreme anterior domain removed, and we can place the green EAD into the unlabeled recipient, and now you've got just 500 green cells in this recipient. And then as the embryo develops, you can watch and see where the cells you transplanted went because they're green, and then you can look at things like face width and the various cartilages and the brain. So here's some of the data. This is probably the most, this is a little hard to get around, but bear with me. It's really fascinating data. So in this set of experiments on the left, we're putting an extreme anterior domain transplant from the donor into an unperturbed, a regular embryo. So if we put a control, a normal extreme anterior domain into a normal recipient, you get a regular head. And you can see here where the green cells went in this head-on view of the embryo. We can also put an extreme anterior domain from an embryo that doesn't have any of this frisbee that we've been studying into a normal embryo. So the embryo is exactly the same as this top one, which gave a certain normalized head width, the regular head width of the embryo. But when we put in an extreme anterior domain from a donor that isn't making frisbee, you can see that the head is much smaller. And that's what these embryos look like. They have narrow heads. You can see where the green cells were. And this tells us that this little group of cells that isn't making frisbee is not able to make a normal head either. We can do it the other way round, where we can, and so this experiment tells you that the extreme anterior domain making frisbee is necessary for head formation. And here is the flip experiment, where we're putting the extreme anterior domain into an embryo, a recipient, that is loss of function for frisbee. So this whole embryo is not making frisbee. And here we've taken that embryo and we've put in a control, a normal extreme anterior domain, and the head actually looks pretty good. It looks pretty wide, the mouth opens, and everything looks okay. If you put the loss of function extreme anterior domain, no frisbee, into a no frisbee embryo, you get this very narrow head, and the various cartilages and things look wrong. So this tells us that the loss of function embryo is not developing normally. It's got a very small head. But if we put 500 cells from a control embryo into the loss of function, now the embryo develops very well and you get a regular looking head. So this experiment was pivotal. It told us that frisbee from the extreme anterior domain was both necessary and sufficient for a normal head size, brain, and cartilage. Okay. We also asked the question, based on our hypothesis, as to, how far the signal from the EAD went. And the way we did this was to use a frog line that was making a green fluorescent protein linked to a reporter that was activated by beta-catenin signaling, the signaling that is inhibited by the frisbee. And the experiment, one of the experiments that Justin did, was to remove the extreme anterior domain from this embryo that made green fluorescent protein in response to beta-catenin. And the results that he got were really quite astonishing. So at the bottom here, you can see here's a control embryo. And let's just look on the right here. This shows the green protein coming from the response, the responsive gene from the embryo, and you can see in the control there's very little expression. It's there, but it's very little. When you remove the extreme anterior domain, you now see a massive increase in the amount of this green protein being made as the inhibition is being taken away from this reporter construct to increase the amount of expression and the amount of green fluorescence. And what these data told us, I won't go through the whole landscape here, but what these experiments allowed us to measure is that the extreme anterior domain was affecting cells way far away. These neural crest cells shown here, but also, I haven't shown in the picture here, but also brain cells, which are very far away from the extreme anterior domain. And we could measure how far the signaling from the EAD went. It's many, many cell diameters, maybe 40 cell diameters from the extreme anterior domain to affect all these cells around. So this told us there was a signal that was traveling all the way through the head. We wondered if the face and the brain were small and the cartilages were small because the cells weren't dividing appropriately. And so we did an experiment to remove the extreme anterior domain. We also did some transplants, but I'll just show you from the removal, which is on the left side of the slide here. We removed the extreme anterior domain, and here's the control embryo. We then stained with a marker for dividing cells, and each of the little dots here is a dividing cell. And when we removed the extreme anterior domain, the number of dividing cells went down significantly, and you can see that in these assays here. And that was true for the extirpation and also for the transplant. And this told us that one of the things that the extreme anterior domain was doing, and this frisbee was doing, were to maintain the right number of cells and the right amount of cell division in the brain. So that was very interesting and gave us a bit of a mechanism of how the extreme anterior domain worked. And then I think the last experiment that I'll show you took that one step further. We thought, well, what else is going on? How does this extreme anterior domain really control face and brain size? And so we looked at a later stage at a number of genes that we thought might be targets for various reasons of this frisbee signaling. And I'll show you actually just one set of assays for a gene called TBX1. For those of you who think about this is a transcription factor that is known from studies in mice to be very important in building the face and building the brain. And so here is a control. This is actually a transplant experiment. We've transplanted a regular extreme anterior domain into a normal embryo, and then later on we've stained it for this TBX1. And here easier to see sometimes in the camera lucidus, here is a big band of TBX1. If you put a loss of function extreme anterior domain transplanted into the control embryo, so you now don't have any EAD frisbee signaling, you see the amount of TBX is very severely decreased, and it's also not quite in the right place. So these experiments and many others told us that the extreme anterior domain was also changing the activity of genes that were coming on subsequent to the formation of the extreme anterior domain. So let me summarize what I've told you in this way. This looks a little bit like the summary I gave you before, but I've added a bit more. I've shown you some data from many, many years of work, really from a project that I started in a way as an undergraduate at WITS, but from many years of work that a region of the embryo, the extreme anterior domain, is a signaling center that is sending out many chemical signals across a wide range of the developing face and the brain. The signals control cell division to ensure that there are the appropriate number of cells to build the face and the brain, and also change the subsequent activity of genes involved in building a normal face, brain, and cartilages of the developing tadpole. And in the case where there's been a perturbation, a removal or a change, a removal of Frisbee expression from the extreme anterior domain, the number of cells that are proliferating decreases, the expression of various target genes changes, the face is very small, the brain is small, and the cartilages are both small and not correctly organized. What does this have to do with people? Well, probably a lot. We actually have a collaboration right now with colleagues in Newcastle who are doing experiments in mouse embryos for us to look at the extreme anterior domain in mouse embryos, and we are looking to see exactly what genes are active there. But we know that this region is present, the extreme anterior domain is present in mammalian embryos and in human embryos as well, as anatomy textbooks have told for a long time. But really we know nothing about human extreme anterior domain. But I'm going to speculate, and I'm going to end by suggesting that this is a very interesting observation that may help to explain really a devastating set of birth disorders that are called the microcephalies. Microcephalies are indicated by small brains, but almost always in microcephalic babies, there is a massive change in the face, in the facial cartilages. So really the microcephalic phenotype is the entire head. It's the brain and the facial cartilages. And that looks very much like the embryos, the tadpoles that I've just been showing you, where when you remove the extreme anterior domain, there's a decrease in the brain size and the shaping of the cartilages is anomalous. We know that from multiple studies, there are certain mechanisms that occur in individual cells of the microcephalic brain that can perturb human brain development, but we suggest that there's another mechanism that's a more global mechanism where you're not going cell by cell to control brain size and shape, but that there is a global way of controlling brain size and shape through extreme anterior domain function. And this, in a way, is quite encouraging, because if one is thinking in the future of how you would address microcephalies, it may be possible to think about a global way to correct the entire brain and the face as it has been affected. And that, of course, is a very much future and somewhat speculative suggestion, but I wanted to share that with you as we close up. So what I've told you today are three things. I told you what the extreme anterior domain is. I told you how the extreme anterior domain leads to mouth formation. And I've told you how the extreme anterior domain is a signaling center that controls the size of the face and the shape of the face, as well as the size of the brain. The work that was done was primarily done by Laura Jaycox. Actually, all of the mouth work I showed you was done by Laura Jaycox when she was a dental and a PhD student in my lab, and Justin Chen when he was a graduate student in the lab, as well as with assistance or contributions from some of the other people who are listed here. And started all this work when I was on the MIT faculty at the Whitehead Institute, and have continued it in my lab at Northeastern. And I'm very grateful to the NIH for funding and to the various Xenopus resources around the world for assistance with various aspects of frogs. And I am very grateful to you for your listening today, and to Professor Ballinsky for his extraordinary contributions to embryology that set the stage for all these studies, and to my dear friend and mentor Barry Fabian, who really was pivotal in my journey here today. And thank you so much to everyone. Oops, stop sharing. There, I stopped sharing. Okay. I'm done.
Thanks very much, Hazel. This is Neville Pillay. I'm coordinating the program, so you might see me now. We can actually take questions. Would you prepare to take questions from the audience?
I would be delighted to take questions.
Good. I'm looking at my colleagues who can raise. Anyone want to ask a question? I see Professor Marcus Byrne.
Shall I go ahead? I'm not an embryologist, so it might sound like a facetious question, but in evolutionary terms, given that the mouth must be a really ancient structure, surely it's no surprise that it has dictated all these other structures downstream of it. So did you start with that as a theory?
Sure, thanks for the question. You know, this is a fascinating question, and we wrote a review article on mouth formation where we really had to work hard to understand what exactly was. I see myself twice. Is there a way to remove me once? It's a really fascinating question to think about where the mouth came from and why. What we concluded when we went way back to sea anemones, which have only two of the layers. They've only got the blue and the yellow layers. They don't even have this red mesoderm layer. We concluded that actually the process of mouth formation probably only arose once in evolution, and that was based on various considerations, including gene expression and so on. But actually there's more. The embryo initially had the blue and the yellow cells that make the mouth right next to it, but as embryos evolved, it's amazing that the blue and the yellow cells that form the mouth actually start off in totally different places. And then as the embryo develops, those two kinds of cells come together and find one another, and that's where the mouth comes from. So that actually blew me away, because it says these mouth cells really know how to find each other and then open the mouth. You know, those are the facts. And so that again shores up that mouth evolution probably was a one-time thing, and it was so useful. You know, you have to get food into a multicellular animal, and really a kind of an essential thing to form. And once it formed, it never could go away, because you can't survive as an embryo without, or an organism without a mouth. You know, whether it's the whole signaling aspect of mouth formation was totally unexpected. The whole signaling aspect of that region of the embryo was completely unexpected. And in fact, the evolutionary origin of mouth hadn't really been traced before. So I would say, you know, this whole aspect of mouth formation and the extreme anterior domain as a signaling process has been very surprising. It didn't have to work that way, but it turns out that it does. So I'm not sure, you know, how to answer your question exactly. I try not to expect anything, but to, you know, get the question asked and answered. And it turns out that there is both this conserved, deep evolutionary process by which mouth formation seems very similar in a sea anemone, at least to some extent, as in a human. But then there's this whole other aspect of signaling that's associated with formation of the face, which, you know, was not appreciated previously. Thank you for the question.
Thank you, Marcus. Can you hear me? Okay. Any more questions? I have one if no one wants to. Zvinka, I'll take your question as the last one.
I really enjoyed, thank you so a lot for your presentation. I really enjoyed the way you're communicating your research. And I was wondering if you have any recommendations for students on how to decide on different ways to communicate your research. Like how you came up with the idea to do this whole modeling from plasticity.
Why to use frogs? Is that what you're asking? Or
no, just the communication, how it was presented, that you've explained how the mouse is formed out of plasticine.
Oh, the claymation! Like the claymation. That was fun, wasn't it? There's a whole genre of movies and things that use modeling clay to make animated figures. Actually, it's called, I can put it in the chat, it's called claymation. And if you look it up, you'll see that there's some very fun movies and things that have been made with little figures that are sequentially animated. And so a couple of students in my research group decided to try to make the formation of the mouth a bit simpler to understand by pulling out just the cells that would make the opening and then depicting those with the claymation. And it was actually quite difficult to do. They sequentially took the modeling clay and changed it a little bit each time, and then they took a sequence of images, of pictures, and then they could link them together to make the movie that I showed you. And that's the claymation. It's requires a little bit of finessing so that you get everything in the right sequence. Thank you for the question.
Thank you. I'd like to ask a question, Hazel. You talked about deuterostomes because you raised that initially in your opening statements. And deuterostomes are notorious by having the mouth developing at a certain time versus protostomes. So you do mention that the mouth is critical in terms of the formation of the head, and you went through the domains particularly. And it surprised me that the entire head region is governed by one region without any checks and balances through the brain after the cascading effect. I think that's what Professor Byrne was starting to ask. So I was surprised that one signaling center decides the entire fate of the head without any reciprocal processes down the line that have checks and balances. This is how it works, a cascading effect from the EAD right through in terms of the head region. Is it right?
I love your question. Thank you. And, let me say, I wanted to get the point across to an audience, and I'm not sure what all of your expertise is. So I really sort of just focused on one region. But absolutely not. The extreme anterior domain is not the only region that is dictating formation of the brain and face. In fact, our own research has uncovered a whole bunch of other signaling regions as the brain is put together, especially groups of cells that dictate different parts of the brain and tell them to form. And then there are a whole set of processes that are also involved in having the neural crest cells move into the face. So I would say, the arrangement of checks and balances during development is quite extraordinary. It's how things work. It's kind of like building, you build one bit and then that allows you to build the next bit and allows you to build the next bit and so on. And there's a lot of conversation between cells during the whole process. So I simplified things and told you just about one region of the embryo that signals, but you should not take that to mean that it is the only region that signals. There is a whole bunch. There are probably 100 different regions of the developing embryo that are all working together to build the brain and to build the face. It's a very complicated orchestration. And I can give you one example to emphasize that. We tried when we started, when we reopened the formation of mouth, the field of mouth formation, we tried to create a mouth elsewhere in the embryo. to see if we had all the genes involved and if we knew how it worked. And it turned out it was very, very difficult to do that. The only way we could get a second mouth was to really take a really large chunk of the head and transplant that somewhere else. And then we could get a second mouth. But we really couldn't do it without a large piece of the embryo that was presumably had a whole bunch of different processes coming along with that. So it's a great question, and I gave you one part of it, but actually the larger question, and I think Professor Ballinsky understood this enormously, and certainly Professor Fabian, instilled this in us even when we were undergraduates, that development is very complex, and it's a wonderful symphony of cells having conversations with one another. And now we know many of the molecules involved in the process. So I want to clarify that one a little bit, but thank you very much for the question.
Yeah, thanks very much, Hazel. The last time I did embryology was in undergrad, so it does test my memory. But I think your talk gave us perspective, and I followed the logic of the argument, so that was the important bit. But I would like to thank you very much for sharing your expertise today in this symposium. What struck me, of course, is how far back the initial ideas started at WITS. You also mentioned thinking about these issues when you were an undergrad, so this goes far back, in some ways mirroring what Ballinsky did when he was a student. So I'd like everyone to give Professor Hazel Sieve a round of applause, and thank you very much for making up so early in Boston.
It's a pleasure, Nadia. It's so good to see you and everyone else.
One question.
Vladimir, you have a question for me? Hello?
Of course. Nice to see you after
Nice to see you also.
quite a few years and quite active in the early hours.
I especially got up. I think this is a really special event, so I set my alarm and I especially got up, and here I am.
Super, so let's get closer to the home ground. Okay. So, it looks to me that this set of experiments is somewhat similar to what performed by Corinne Huard on zebrafish with analysis of anterior neural reach and TLC1 as a Wnt inhibitor. And I wonder what this sort of connection, whether it's the same area or, of course, she did not really look at cartilage, but only on neural structure.
Yeah. So as you know, we've done a lot of work in zebrafish development and brain development. Karin pulled out that very front region of the, we're getting into deep embryology, everyone here. Karin pulled out the very front region of the anterior neural plate and showed that there was a signaling center right along that border at the time the brain was putting together. It's a different region than the extreme anterior domain, which is actually very hard to study in zebrafish. It's a very small region in zebrafish, not a good model to study it. It's there, and we've actually published work on gradient kinase signaling, which is another nitric oxide signaling, another pathway that's involved. But it's not the same region that Karin looked at. The extreme anterior domain, since you and I are now having a little embryological conversation, actually is probably ventral to a region called the FES, which has been described in chickens, which expresses sonic hedgehog and FGF in a kind of stripe. And so what we've shown, what I didn't show today, is that the extreme anterior domain is a very early domain that induces the FES to form above it. And Karin's region is sort of, more posterior to that as well. But yes.
Okay, thank you.
Okay, thank you very much. We'll leave Professor Sieve there, and we'll move on with the rest of the program. Thanks again, Professor Sieve.
My pleasure. Thank you for having me.
So if you follow your program, we now move on to Zvinka Kacho, who I met at WITS a few times, maybe twice. So she's a regular visitor to us. She represents the Ukrainians in South Africa society, is that right? And you might see their banner outside. And she'll speak a little bit about her own research in Ballinsky, and I know that we've made available some of our resources in the museum that you've seen, Zvinka, but you might have added to that as well. So welcome, Zvinka, to the symposium.
Thank you. I'm very honored to be here, and I'm very humbled to speak after this excellent scientific presentation, because my talk will be more about the Ukrainian community, so that you can better understand how Ballinsky fits here, and also how Ukrainian community is shaped today. And because we treasure Ballinsky as one of the symbols of Ukrainian community, the connection between our two countries. So I'm just going to move to my presentation. I think I'm right. Oh, no, I'm not. No. Okay. Maybe I'll get the stick. Oh. I apologize for that. We did try this presentation earlier, and it was opening, but I can start you from while we are sorting out the technical matter, maybe to tell you a little bit about Ukrainian Association of South Africa. It's a non-profit organization registered in 2017, and it unites both Ukrainians and South Africans. So if you are interested, you obviously are welcome to explore those connections. Oh, thanks. Do you have the thing or not? Thanks. Sorry. Okay, here we go again. So that's about Ukrainian Association of South Africa, and I wanted to show you where Ukraine is, because while we are quite far away, and I'm not sure how many of you actually went to the map and check where Ukraine is, and Ballinsky's hometown is Kyiv, where I'm also coming from, where my family is today. So Ukraine is in Eastern Europe. And there are about 42 million of people. It's one of the largest country in Europe with 600 square kilometers. And more than 50% of population have higher education. So this tradition of higher university degree is very popular. Ukraine also is one of the founding members of United Nations. So it's a country with long history, and it received its independence from the Soviet Union in 1991, and that's when the diplomatic relationship between Ukraine and South Africa have started after Ukraine became an independent country. And this symposium was also partially initiated to celebrate the 30 years of diplomatic relationship between Ukraine and South Africa. If you are wondering how many Ukrainians are in South Africa, there are about 5,000 to 6,000 based on data on 2020. Also, the number of South Africans is not very big, and they are mainly concentrated in Johannesburg, Cape Town, Durban, and Quebec. We can see that the amount of Ukrainians have increased between 2014 and 2015 when the initial invasion of Ukraine happened, and it's linked to 1.5 million of Ukrainians becoming internally displaced people already in 2014, because the Russian invasion has happened not in 2022 but in 2014. So some of those Ukrainians came also to South Africa. A majority of Ukrainians play a role in the economy in South Africa. We have quite a few doctors that came here from early 90s, but also more modern IT technology and business development and private entrepreneurs. Those are predominant occupation based on the research that we've done on Ukrainian community in South Africa. If we look on the history, how Ukrainians migrated to South Africa, we can think about the first migration that happened already at the end of 19th century, beginning of 20th century, as the Russian Empire moved the line that prohibited Jewish communities to live in cities, also in Kyiv. Those Jewish people were pushed out of cities, and many of them migrated to South Africa. It's estimated that about 15,000 of members of Jewish communities moved from the territory of Ukraine to South Africa. And those of you who like music by Johnny Clegg, in his autobiography he mentions also that his grandma was coming from territory of Ukraine. There was also another way when Ukrainians trekked all the way to South Africa during Anglo-Boer War, because there was a lot of sentiments to support Boers, and one of Ukrainian poets, whose real name was Yuri Budyak, he describes the stories of his adventures in Anglo-Boer War, but the majority of those volunteers, they would go back to Ukraine and didn't settle in South Africa. So the individuals from Ukraine were coming to South Africa as a result of the First World War and Second World War. So predominantly these were refugees who could not stay in their home countries because of the different kind of prosecutions. And this is also a story of Boris Ballinsky, who came to South Africa because he could not remain in Ukraine. There is also another chapter of our joint history. It's Ukraine and anti-apartheid movement. Soviet Ukraine, being one of the 15 countries of the Soviet Union, has trained ANC cadres since as early as 1963, and actually the first training camp was established in Odessa, but also many other representatives of anti-apartheid movement received their higher degrees in universities in Ukraine, medical degrees, engineering and chemistry, environmental scientists also. So that connection also resulted in some of the South Africans coming back with Ukrainian families, like Sindiso Mfenyana, who in his autobiography mentions that Kyiv is his second home, as he spent lots of time studying and living there. And Soviet Ukraine was also played a big role in anti-apartheid movement. It was on the anti-apartheid committee in the United Nations, and is co-sponsor of many resolutions, including the Sharpeville resolution. And the last fun fact about Ukraine-South African connection is about the geographical location, Mazeppa Bay in eastern London, in eastern Cape. There is a geographical point that is linked to one of the leaders of Ukraine, Ivan Mazeppa,
who was the head of state at the end of 17th century. I can assure you he have never been to South Africa and has never colonized anybody, but his love and passion and fight for Ukrainian independence became the symbol in Romanticism. So many authors, including Byron, Voltaire, wrote their poems about Mazeppa. So the name Mazeppa became the symbol of speed, fastness, and freedom, and it became popular to name different boats by the name Mazeppa, including Byron's boat was named also Mazeppa, and one of such boats was built and then traveled along the coast of South Africa and gave the name to this geographical location, a beautiful bay that is now called Mazeppa Bay. But we really treasure that Ivan Mazeppa has traveled not in person, but in spirit all the way to South Africa. There is also a street called Mazeppa in Durban. And South African poetry, the new Romanticism wave at the beginning of 20th century also used this image of Mazeppa, and Roy Campbell wrote a poem dedicated to Mazeppa as well. But in South African literature, if you look, Mazeppa, it could be the Zulu king or Polish king, so the identity changes, but the name remains, and the spirit. The tiny museum in Riversdale represents some of the Ukrainian culture, and also over 20 years of cooperation between the Riversdale and the Ukrainian city of Voznesensk. So people were traveling to share one another. All that is written in our little study, Ukrainians of South Africa, which is available online, free. So those who are interested, please visit the webpage. We also had a conference to explore the formation of some other Eastern European communities, Bulgarian, Hungarian, Polish, Romanian, and that research is also available electronically on the webpage. I personally very grateful to Boris Ballinsky for writing down his memoirs. He wrote them in English, and when I happened to read them, I think the electronic copy was in Illinois University, so it was quite surprising to find this connection between Ukraine and South Africa, and now Illinois Library. And here is this autobiography tells about very difficult time in Ukrainian history, but also the time that has been so manipulated that there are very, very few witnesses who are telling with lots of details on what was happening in Ukraine, but also very accurately, without manipulating different events. So, for example, Boris Ballinsky mentions that when in 1917 Ukraine declared independence, his family celebrated it. Already a few months later, when Bolsheviks took over Kiev, parents crossed that line so that nobody can prosecute them for supporting Ukrainian independence. Ballinsky also witnessed Holodomor, a man-made famine that took lives of 3 to 9 million of Ukrainians. This famine was organized by Stalin in 1932-1933, and for those who will travel to Cape Town, there is a memorial bench in the Val Park dedicated to all victims of Ukrainian Holodomor, and also established in solidarity with the victims of genocides on the African continent. And it says, for the sake of a sustainable future, we have no right to forget. And I hope that getting back and exploring the legacy of Boris Ballinsky will also inspire us not only to learn about the science, but also about how we can continue doing science in the most difficult situations. And I hope our colleagues in Ukraine can also hear me on that. I think Boris Ballinsky also represents that missing middle. he emigrated. And we were discussing here that, is he Ukrainian? Is he South African? I think as a Ukrainian, I'm very grateful to South Africa that Boris Ballinsky could flourish here because in Ukraine, the whole institute, that was leading institute in this research, has been eliminated. More than 3,000 scientists who were researching genetics and embryology were either prosecuted or killed due to the predominant theory that genetics is not a true science. So at least now we can rediscover Boris Ballinsky. As you've heard, his book was translated to many, many languages, but never to Ukrainian or Russian or any other Eastern European language. And the reason is that he was forgotten there. So now we are trying to rebuild the trip back for Boris Ballinsky. He himself did this trip back in 1996 after South Africa and Ukraine both became independent. Boris Ballinsky twice traveled to Ukraine, and he also donated his beautiful collections of butterfly to his first alma mater, Kyiv University. One of the inspiration behind the scientific research for Boris Ballinsky, as he describes, was his grandfather, who encouraged him to research the frogs when he was still a young boy. And his grandfather, Vasily Radzimovsky, was murdered by Bolsheviks during the revolution when he was 72. This photo is from the family album. Thanks, Helen. And in March 2022, Irpin, that hometown where Ballinsky spent most of his summers, became one of the places for mass killing of civilians by the Russian army. Unfortunately, 290 civilian bodies were discovered as the city was liberated after the occupation. Among them was a 70-year-old professor who was shot in front of his wife. Those are sad stories, but the story of Ballinsky tells us that science continues, and we hope that many, many new researchers will be encouraged from this event today, but also from reading the stories about Ballinsky, hearing this beautiful scientific presentation like we had heard today. So thanks a lot for having this symposium and also allowing this cooperation between Ukraine and South Africa to happen. And those who want to become a member of Ukrainian Association, you are welcome to follow us. Thanks.
We need to get Helen David's book. Take this way. Thanks again, Svinka. We met on a few occasions, and I remember you telling me bits and pieces of the story. And Anastasia, maybe you gave me books on the famine that was so devastating to Ukraine. But it's good to see you put the whole story together. And you did mention on a number of occasions the Ukrainian South African Association and the work that you do around the country. So thanks again for that. I mean, now I have the privilege of having Helen David speak to us. And those of you, I think we've heard a number of times already today, Helen is Boris Ballinsky's daughter and a BITS alumnus. I think Helen was from physiotherapy, is it right? So, and she's a regular contributor to Yerbo Kocha. So I learned that subsequent to dealing with the Ballinsky Symposium. I didn't know that before. So hopefully we'll see you this year again, Helen. But over to you.
Thank you very much. Thanks very much. Are we okay? Just to take it that way a bit, please. Thank you very much. Good morning, everybody. I've been asked to give you a personal view of my father, Boris Ivan Ballinsky. He was a very special man, a multifaceted man, and I hope that this presentation will give you some insight into his life. I will be speaking of the years from 1949 to 1997, which was in effect the second half of a long and full life. It's the years in South Africa. My father left Europe accompanied by his mother, and his second wife, Elizabeth, whom everyone knew as Betty, his teenage son and his baby daughter. And they arrived in South Africa by ship in Cape Town to start a new life. This is an image of my father as he is in my mind and my heart always, full of warmth and full of wisdom. In those days we lived on what was then the northern outskirts of Johannesburg. Here is my father with his two children, Ivan, or Vanya as we called him in the family, and myself. And this is our grandmother, Elizabeth Radzimovsky, who died when I was 4 years old. Here is the family in Blegari. There was a 15-year age difference between my brother and myself, but we were very close in spirit, especially when we were both adults. My father worked hard in the quarter-acre plot of ground to create a garden. He was a strong man, mentally and physically. He had to have been strong to survive the years of oppression, of war, of displacement, of loss of the people and the places that he loved, and to start a new life again in a new country with energy, with enthusiasm, and with a positive attitude. Gardening was a much-loved activity throughout his years in South Africa. Boris planted fruit trees such as peaches, figs, apples, pears, plums, and indigenous trees, and he grew strawberries and grapes, which we all enjoyed. In his later years, even in his 90s, when we visited him, we often found him sitting or kneeling on the lawn and weeding, an activity which he found to be therapeutic in times of trouble and stress. I also wanted to tell you about the rockery. He built a rockery, digging up and moving large rocks by himself. This is our street, Oban Avenue, looking across towards where Sandton City now is. Can you believe it? Now then there was nothing there. And the current Delta Park is also down in the dip there. My father would set off on his bicycle, collecting nets strapped to his back, and go down to the ponds and the streams, collect butterflies, dragonflies, and the eggs from frogs that he needed for his experiments. And sometimes my brother or I or both of us accompanied him. This is our street, a gravel road. Our house is on the left. It was one of the few houses in that area in those days. And milk was delivered by donkeys on a cart. In our back garden we had a few chickens to provide eggs. My parents had very few possessions after the war years, really very few, and life was very basic. This is our home many years later with a mature garden and beautiful trees. In 1997, and my mother was then moved out of the house and died 2 months later. The Witwatersrand University was a huge part of Boris's life for so very many, many years. And both his children studied here.
I did a BSc physiotherapy, including first-year zoology, and was thus in my father's class for some lectures, which was quite interesting. He did not say one word that was not important. We had to take down every single word. And in addition, he drew beautiful diagrams on the board in chalk behind him, of all the black and white sketches are done by himself in black ink. And they're very, very precise and good. John, as he was known in South Africa, did a BSc with majors in chemistry and zoology and honours in biochemistry. Both of us became lecturers in our fields at Wits and elsewhere. For Boris, work was a pillar of his life. Here he's seen in his office in the zoology department. My father was seen differently by different people. Some found him stern, serious, and even formidable. And there's no doubt that he held very strong and sometimes very stubborn opinions, which he was not afraid to voice. Others found him relaxed, warm, friendly, helpful, and very generous. My father spent many, many years of his life in South Africa working on this book, An Introduction to Embryology. There were 5 editions, several reprints. It was translated into several languages, and there was even a posthumous Asian edition. The 100,000th copy of this book was celebrated at Wits University, and here he is seen with Professor Bozzoli and with Betty. My father formed many close relationships with colleagues over the years, and there were some very special friendships. Here you see him with Barry Fabian, absorbed in research work and at the inauguration of the B.I. Ballinsky Laboratory, which was named in his honor. Family was the other pillar of Boris's life. Here is Vanya with his baby son Andrew, and the Johannesburg of the 1960s in the background. It looks very different now. Vanya and Doris, with their sons Andrew and Martin, lived in Parkmore until they moved to the USA in 1975. The love of an African adventure was common to all our family members. Doris and Vanya took their sons Andrew and Martin to the bush, to the coast, to the mountains. Elaine and I took our children, Christoph and Danielle, to the bush, the coast, the mountains, the deserts. Dogs were an important part of home life. My father walked with them daily in the streets and found comfort and joy in their company. Exploring Africa was a great joy to Boris. In the early days, in our first car in Austin, we used to go on the weekends to nearby places such as the Magaliesberg for walks and for picnics. And we laughed at my father's often said comment, "Once I see a monkey, I really feel I'm in Africa." Here you can see his love of nature, relaxing on a holiday in the Drakensberg, no doubt after a successful outing to collect butterflies or dragonflies. He loved to climb mountains, as he had done in the Caucasus Mountains in his youth. He climbed Champagne Castle, which is well over 3,000 meters in height, at 62 years of age. This was quite a hike, climb. It took 16 hours, and he started and ended in the dark. And although he did not quite reach the summit, he was given a welcome on things. Here you see Cathedral Peak in the center, with Champagne Castle peeping out at the back. There were several adventurous trips in our Land Rover to the Okavango swamps in Botswana. The Okavango River originates in Angola, enters Botswana, and then divides into a network of channels of crystal clear water, which disappear into the sand of the Kalahari Desert without ever reaching the sea. This unique and beautiful area was captured in In those days, we camped in a canvas tent. There were no official camps, no official campsites, no fences, and animals. Sometimes we woke to find very fresh lion tracks surrounding our tent just a meter or two away, and we never knew what was behind a bush or round a corner. This is our cottage at Manzimvula, a small private game reserve adjacent to the Kruger Park, where we spent many happy times. This photo was taken in 1970, but the story began some years earlier when a gentleman spoke to us and said, "How would you like to own a place in the bush with a giraffe and other animals on your doorstep?" Well, Boris was hooked. Even as a child, excuse me, even as a child in Kiev, he had dreamed of going to Africa with its spectacular variety of wildlife. And as a zoologist, the diversity of Africa's wildlife was a huge attraction. Here at Manzimvula and in the adjacent Kruger National Park, he could observe hyenas and other predators such as lions, leopards, jackals, interesting small mammals such as genets, bush babies, squirrels and mongoose, and the larger herds of buffaloes and elephants, as well as a variety of antelope. Insects, birds, amphibians, reptiles, all creatures great and small were of interest to my father. Manzimvula was a place to enjoy time with family and special friends, a place to inspire yet another painting, this one of a blue wildebeest which hung in our lounge for many, many years. Here is a classic photo. Insect collecting net in hand, walking in nature, a large grin on his face. My father was a real collector. He turned over stones in riverbeds to find stoneflies. He swished his net along streams to catch dragonflies. He set traps with ripe bananas to attract butterflies. And he left a lamp out at night to attract moths. In the garage of our home, he made his own insect display cabinets. He was a competent carpenter. Here you see one of the display cabinet drawers with Acrea butterflies. In his 70s and 80s, he conducted meticulous genetic experiments with these Acreas, and he found this to be a source of pleasure and of satisfaction in his years of retirement. Boris loved to travel to different destinations in Africa. Victoria Falls and the Hwange National Park in Zimbabwe were memorable destinations. There were also trips to Namibia, Malawi, Mozambique. There were several trips to the Marine Biological Station on Inyaka Island, just off the coast of Mozambique, with students and staff from the Witwatersrand University. And sometimes the family tagged along and learned about marine life. This is a painting of the western flats, which teemed with fascinating marine creatures. My mother and I would stroll along and I would learn about all these fascinating creatures from the student scientists who were there. Trips during Boris's later years included the beautiful Blader River Canyon in Mpumalanga, and perhaps his most adventurous trip of all, a cruise to the Antarctic, which both my parents found absolutely fascinating. My father was multifaceted. His hobbies and interests were many and varied. He grew the classics, once commenting that Dante's Divine Comedy was difficult to read, and when asked, he was reading it in an old form of Italian. He read modern novels, poetry, scientific material. He loved the Scientific American publication and newspapers. He spoke to a better or to a larger or lesser degree 7 languages by astronomy, and we had an amateur telescope through which we observed the moon, the stars as a small. My father was a complex man, but at the same time a simple man. He enjoyed simple pleasures such as a home-cooked meal, a glass of wine, and good conversation. Here he is seen with my husband and myself, aged 90, still alert, still interested in a variety of topics. My parents looked after one another throughout the more than 50 years of their married life. For the last 5 years, my mother was in a wheelchair and my father took good care of her. This photo was taken in 1978, but the quote comes from notes that my father made in the last year of his life. In spite of all troubles, I am still a happy man. My father left huge footsteps, huge footprints, and he would be honored and delighted to know that there are many following in his footsteps, both personally and professionally. Thank you so much.
Thank you, Helen. A moving tribute to a great man. We're going to move on with the program so that we can meet the lunch deadline. But again, we move back to the scientific program. Is there anyone that has to leave for various reasons? I know there's a talk happening in the school. You might want to do that now or maybe wait a little bit. But if not, we'll continue with the program. We move on to the scientific part again. And Professor Vitaly Karchenko from the Schmullhausen Institute of Zoology in Ukraine, which where if you remember the earlier conversation, the Schmullhaus was one of the people that inspired Ballinsky to go and collect his collecting trips. I think mainly in the sea urchin part of his collection, but I may be wrong. Ballinsky Institute is going to speak about the Ballinsky Institute at the Institute of Zoology in Kiev. I think it's an online feed into the venue. I can't see if anyone's here. Okay.
First of all, I sincerely want to thank organizers for the invitation. Ukraine is a very rich country that allows itself to give its best sons and daughters to other countries without slightest regret. Here is it called our gift for the world. Among many others, Boris Ivanovich Ballinsky was such a gift. I did not know Ballinsky personally. I did not meet him even during his visit to the National Science and National Museum or National Academy of Science in Ukraine in 1993. Actually, I learned about him from Alexei Humovsky's report at the Academic Council of our institute. However, I am sure that Professor Grigory Dvoinos and me were among the first who established relations with South African Republic in 1991. The world knows Boris Ballinsky as Ukrainian and South African biologist, embryologist, entomologist, professor at Kiev National University and Witwatersrand University in Johannesburg, a pioneer in experimental embryology, electron microscopy, and developmental biology, the author of the world famous textbook Introduction to Embryology. Boris Ballinsky lived in South African Republic for about 50 years of his life. This report will similar to the critical article A Ray of Light in the Dark Kingdom by Nikolai Dobrolubov, 90% of which he used for highlighting his own political view rather than discussions the play The Thunderstorm by Alexander Ostrovsky. I realize most likely you don't know who is Dobrolubov, but artificial intelligence told me about similar work in English literature, such as On Liberty, John Stuart Mill, published in the same year. Boris Ivanovich Ballinsky was born in September 10, 1905 in Kyiv for 5 years. 1917 and 1923 he studied at school. When father asked son how he see his future, the boy told he wanted to work at zoological museum. If so, be ready to wear patched pants for the rest of your life, answered father. Since then, wellness of young Ukrainian zoologists has changed very small. In general, his childhood was like in young naturalist. In 1962, my parents took me to see movie Drums of Destiny with George Michael, and that might be push me to choose my life path. Boris' mother, Elizaveta, was a biologist. To make and meet, the young man had to popularize science. As many talented people, his interests were diverse. In 1926, Boris graduated from the Institute of Public Education. That name, the name of Taras Shevchenko Kyiv National University, and entered the graduate school at Fedor Ovcharenko Biological Institute. In 1933, at the age of 28, he was already a doctor of biological sciences and a professor at Kyiv University. About Ballinsky works at Institute of Zoology we will speak later, because it was founded in 20s of the previous century. The Biological Institute of All Ukrainian Academy of Science, also known as a Microbiological Institute, Fedor Melchenko Biological Institute is a scientific institution that was in Kyiv in 1920 and 1930. Created as private laboratory of microbiologist Melchenko, later became Ukrainian Scientific Society, and upon its liquidation, the second department of All Ukrainian Academy of Sciences. After the death of academician Melchenko, the institute took his name, and academician Ivan Shmalgausen was its director. In 1930, the institute became one of structure laid in the basis of the Institute of Zoology and Biology of All Ukrainian Academy of Science. In June 1918, Melchenko moved his laboratory from Petrograd to Kyiv. This laboratory was located right in Melchinsko's apartment, and he donated all its equipment to the Ukrainian Scientific Society. Ukrainian Scientific Society adopted the statute of Institute of Microbiology, where Department of Zoology, Botany, Bacteriology, and etc. were to be created. However, the lack of funding left this plan unfulfilled. In 1924, after the death of Melchenko, academician Ivan Shmulgausen was appointed as director. In 1926-27, in addition to director, the staff of Institute of Biology include Skovoroda, Zachinaev, Bordzilovska, Singaevska, Ballinsky, Brust, Dragomiro, and Lipsky. In 1919, the newly created All-Ukrainian Academy of Science established a number of its institutions, including Zoological Museum of Ukrainian Academy of Science. According to decision of Committee for Studying Fauna of Ukraine, Ukrainian Academy of Science Zoological Museum was created. Academician Kashchenko headed the committee of later museum itself. That time zoology of Kashchenko, Paramonov, Karavayev, Voskoboynikov worked here during 1919 and 1923. The future outstanding evolution biologist Dobzhansky joined them. Thanks to further development of zoological research since 1926, a powerful team of zoologists led by Karavayev has been formed. For 2 years, the museum occupied 2 rooms in the building of Academy of Sciences. Here is on Cross Volodymyrska Street and Rarezna Street. Boris Ballinsky made remarkable sub-researches in triton embryology. In all, I performed in this first year about 130 operations, and of this only about 8 survived long enough for my purpose from the autumn 1926. In addition to scientific work, teaching at the university, he was also the deputy director of the Zoological Institute. When I was appointed first, the 15th of June that year, 1936, I was wondering how should proceed in my new administrative capacity. He occupied this position for 1.5 years. I was the deputy scientific director at the institute for 19 years and wanted to know better about this side of activity. However, Boris Ivanovich's memories did not cover this area in detail. After the arrest of his wife in 1937, Ballinsky left his position in the end of this year, still working at the institute. He was 32 years old. After release, Ballinsky's wife in 1939, the situation improved a little. He edited some monograph, received very attractive offer from Moscow and accepted them. Pardon. After his immigration, the name of Boris Ballinsky was banned, and by rules of that time, his book had to be removed from libraries and destroyed. However, Scientific Library of Smolkhausen Institute of Zoology has some saved printed book, and here you can see them. This street is named in honor of Professor Ballinsky in Kiev. Today, Institute of Zoology bear name of Ivan Ivanovich Smolkhausen. This is world famous zoological institution with 9 scientific department. The institute employed 122 scientists. The Ukrainian page in Wikipedia with Ballinsky biography is largest as compared with to English and Russian one. In such a way, Ukrainians try to honor the memories of their compatriot. On photo, Boris Ballinsky sincerely smiles, as always in his life. The report used the diaries of Boris Ballinsky, the article about him in Wikipedia, and article wiped from the history Who is Boris Ballinsky by Alexei Boldyrin. Thank you.
Thank you, Professor Karachenko. Just give me 1 second. Is Kurt around? There's a last speaker before we break for lunch. I just wanted to see if we can technically sort out something. Thank you. So what I was trying to do is that we don't actually get to see the speakers that are presenting from abroad, and it would be nice to see a face rather than a presentation. So we'll try to see if we can, but then of course we need to get them to open their cameras on that side. But we'll do that in the future and see how we go. So our last speaker for the session is Professor Yuri Salia from the Institute of Animal Biology. He's the director there. And he's going to talk about Boris Ballinsky from Ukraine to Africa and back, a century-long journey. Over to you, Professor Salia.
Good morning for everybody. I'm extremely glad, dear participants of the conference, to be with you. Unfortunately, only online, not really. But today, it is a difficult time in Ukraine, the time of the war, and I want to tell you today my talk named Boris Ballinsky from Ukraine to Africa and back, a century-long journey, talking about Boris Ballinsky, about famous Ukrainian and South African scientists. But a couple of years ago, in Ukraine, this famous was known in most countries of the world in Ukraine, his name was forgotten. His name was very never know who is this. Even biology students didn't hear about Boris Ballinsky. It was made certainly not simple. It was made by Soviet power. They didn't want to speak about such people because his difficult and dramatical biography. And Boris Ballinsky way from Ukraine, here I show here, this trip was very long. His trip, I think, was started in his childhood because, as he wrote in his memoirs, he, from his childhood when he was a young boy, dreamed, like most, I think, boys dreamed to visit a nice continent, African continent, to visit Africa. But first years of life of Boris in Ukraine were quite happy. He was born in rich, good family. They have very nice conditions of life. But this time wasn't long. And after the beginning of Russian revolution, after this time was very changed all the life of millions of people in Ukraine, including Boris Ballinsky. And he was, during his first part of his life, he found the way to go at first for Germany through Poland, after that to England for a short time, and after that to South Africa. And my when I was a young scientist in the beginning of this century, in 2002, I also have a trip from Ukraine to Africa to take part in the 15th International Congress of Electron Microscopy. It was took place in Durban, and I remember this visit for all of these years, and I was impressed very much about my visit to the South Africa. I loved this country from the first moment that I came from the plane, and I continue to love your country today. After my first day at the congress that was held in Durban International Congress Center, was the ceremony of opening, and I was impressed once more that a huge number of participants, more than 1,150, and all of them were listening to the first plenary lecture to the ceremony of opening, and the second plenary lecture was made by Aaron Clapp. He was on this photo with me, young this time, the Nobel Prize laureate, famous electron microscopist, and this lecture was Boris Ballinsky lecture named for the memory of Boris Ballinsky. when I listened to this lecture, I was very interested who is Boris Ballinsky, because we never we never know anything about him. And after this lecture, I asked organizers to give me some information. I tried to find some materials about Boris Ballinsky in Africa, and this is photo as well as biographical information that was kindly provided to me by organizers of the Congress directly from the memorial poster dedicated to Boris Ballinsky. This poster was in the hall, the Congress hall, and I take this photo, this original I have for this time, and take it with me to Ukraine. After my return home, I tried to find any information about Boris Ballinsky in Ukraine, dictionaries in Ukraine, encyclopedia in Ukraine, books. Never was any mention about this great scientist. And only thanks to this information that I received from South Africa, I began to write some articles in newspapers to give for Ukrainians information about this famous scientist. And I published some articles in local newspapers, in magazine of Academy of Science, and in other newspapers for students, where I described the biography, the working, the scientific way of this famous Ukrainian scientist. Also, it was first good attempt, because it wasn't very simple to publish such things, because a lot of newspapers or other mass media, they didn't want to publish information about science, They told me that it isn't interesting, and many such my papers were rejected. But that time was very famous, very popular newspaper, Ukrainian newspaper, the Mirror of the Week, and they published large my article about Boris Ballinsky, and after that, a lot of people in Ukraine received information about the scientist. A few years later, after this Congress, I meet in Kyiv on the scientific meetings, young neurophysiologist from Kyiv, Alexei Boltov, and we discussed popularization, question of popularization of science in Ukraine. We discussed the study of the history of Ukrainian science. He accidentally mentioned Boris Ballinsky name, and it turned out that Alexei has a great interest in the scientist, and he made a lot, also a lot of efforts he paid to return the name of Boris Ballinsky to our country. He will be with us on this conference tomorrow, and I think he will give us more new interesting information. And when we started Boris Ballinsky biography, we received also from Illinois University Library the copy of memories of Boris Ballinsky. And speaking about these memories and about the popularization of science, I want to show you some information from his memories. He wrote, while continuing my scientific work, I got involved also in writing for the popularization of science, Ballinsky wrote. The opportunity was provided through my mother's connections in the teaching circles. She was offered to write little popular books on science. Two of these were herself. One was entitled Boris Ballinsky, the other Spring Flowers, was with botanical contents. The third book was Instinct and Reasons in Animals. For the writing of this book, she invited me as a co-author. We both had to look up the relevant literature and produced a little booklet, which I think was quite well written and up to date. All the books were in Ukrainian, they were duly published. The last book under joint authorship of my mother and myself, my mother in India for Elizabeth and B for Boris. So Boris Ballinsky also paid great attention for not only scientific work but about its popularization. And I think it's very, very important for all of us. And when we read Boris Ballinsky memorials, we can find that his life, especially his life, his young years, his first period of his life was extremely difficult. His life was in the time of revolutions, of wars, and we can read in his memories and so they are more devoted to the difficult political and military events in Ukraine in the first half of the 20th century. Ballinsky mentions a lot about Kyiv, about Irpin, about Kharkov. In Irpin, their family lived during summers, and there were troubles in 1890-20. Then a successful struggle of Ukraine with Russia for the independence of Ukraine. He describes the terrible crimes of Soviet communists that they committed there. He described the Holodomor, also known as Ukrainian famine, man-made famine in Ukraine from 32 to 33 years that killed millions of Ukrainians. And the scientists also write a lot about the period of Second World War. And when he reads the names of the cities, Kyiv, Irpin, Kharkiv, I think many of you know that today, last year, this year, these cities also are under the war, our country under the war. In this picture you see the photo of 2022 when Ukrainian citizens evacuated from Irpin because their homes destroyed by Russians. And when we compare these historical things with events described by Boris Ballinsky 100 years ago, they are very, very, very same, unfortunately. But fortunately, our conference today finally, I think, returns the name of the famous scientist Boris Ballinsky to Ukraine. But unfortunately, today he returns to Ukraine, which is again in war. But we believe in Ukraine's victory over Russian aggressor and seek peace. I wish peace and harmony to you, beautiful country, and for all Africa and the world. I know that we have good connections, scientific connections with South Africa, and I think that such initiative that you made on preparing and home such conference. It's a very good idea to continue and not to stop today, but I propose to make such Ukrainian, both literal Ukrainian, African conferences regularly, maybe once in 2 years, and I think that one of them may be in Africa, one of them may be in Ukraine, because I hope that war will be finished and you will have good possibility to travel for our beautiful country. In the end of my short talk, I want to say that I am grateful for Ukrainian army, which today is heroically fighting for the enemy and gives us the opportunity and possibility to work and speak before you. I'm grateful very much for you, all for you, for your attention and for your excellent holding of the conference. Dear Bonga. And last slide that I show you, last picture, this is picture of my city, Lviv, and we are more than approximately 10,000 kilometers from South Africa. And lines are not, we haven't lines in Ukraine, but Lviv is a city of stone lines. We have a lot of stone lines, symbolize our city. So we have many connection with your nice country. I wish you all the best and thank you for your attention.
Thank you, Professor Salia. It's a great tribute to Ballinsky. We have real lines, so hopefully when we eventually have the conference in South Africa, we can have it as a shared experience between Ukrainians and South Africans. That brings us to the end of this session, and we now will break for lunch. But thanks to everyone who's spoken in the last session. I see some of the people still here. Please join us for lunch, and it'll be served outside. Thank you very much. Afternoon, everyone. It is my great privilege now to introduce Professor Denise Scher, who also comes out of the Wits stable under Barry Fabian. And Denise is now at the Barts and London School of Medicine and Dentistry in the UK, and she's going to talk about the molecular determinants of the cell fate in children's brain tumors. Denise, finally, thank you very much for making yourself available. It's been a long journey to get here, and I do appreciate your patience over many years to offer a presentation at the symposium. Denise.
Thank you so much. Thank you very much for inviting me. It is an enormous privilege for me to be here, and it does feel like a full circle in my life, because Professor Ballinsky was so important to me and to determining actually the whole direction of my research and how I've tried to pass on the excitement of development, of the process of development, which has to be, I think, the most magical of all the sciences, the transformation of something from, the little ball of chemistry and physics, the cells. So I'm Professor of Human Genetics at Barts in London, which is the medical school of Queen Mary University of London. I'm also a research lead for genomics and child health. This is I've just put up an image of this beautiful institute that we work in the East End of London. And I'm going to take you through let me just see how to progress this, through my excitement about having worked with Professor Ballinsky and Professor Fabian. And Barry Fabian, in fact, has been a mentor for my entire life since I first met him at Wits. So I was at Wits from 1969 to 1973, where I did zoology and embryology as a BSc first, and then I did a BSc honours in zoology, and I did a project on embryology, which was co-supervised by Professor Ballinsky and Professor Fabian. And I have put up this beautiful picture, which I think you've already seen, of Professor Ballinsky, as I remember him. Warm eyes, always smiling, a sense of generosity about him. And his passion as a scientist, I think, is something that I've wanted to emulate and to be and to inspire others. I've put here images of these two books. Let me just put the laser pointer of these two books. And these are my original books that I have on my desk. I've got the other editions as well, but I just thought I'd put these two up just to show how inspirational they've been and how they've carried me forward over the years. This is how I remember Professor Ballinsky and Professor Barry Fabian, always busy with some system or other that they were investigating. The chick development is something that we started out with when I was a student. I used to go to Bedford View to go and pick up my little batch of eggs, come back, put them in the incubator. And I was taught by Professor Ballinsky and Barry Fabian how to dissect, how to keep the embryo growing. And I just want to say that throughout all of this, it was Barry who took me to the next stage of thinking ectoderm, mesoderm, endoderm, and ultimately to the fact that all of these beautiful processes that take place during transformation, that DNA somehow, for me anyway, underpins all of these beautiful processes and what confers the phenotype. And of course, it's the interaction of the environment, even just the environment in the sense of the cytoplasm and the other cells and broader environment, into transforming the basic tissues into organs and then ultimately into an organ. I'd also like to remember Professor Ballinsky for his inspiration not only in development, but thinking about evolution all the way through. And I know that he was greatly influenced by Dobzhansky, and he often spoke about Dobzhansky's that nothing makes sense except in the light of evolution.
And somehow the trips to Inyaka Island that we used to go on, and this is a copy of my book of the natural history of Inyaka Island, and Boris Ballinsky was directly involved in producing this book. But all the time we were made to think of biology as evolution. This is a beautiful image of the chick in development. And of course, what worked in my mind and for all of us all the way through is the magic of how a limb could be induced, as shown by Professor Ballinsky in 1925, by the transplantation of the ear primordium into the side of the flank of the Triturus salamander to generate a limb. And this process of experimental induction is something that, again, is an embryo of an organism. These are a couple of pictures taken from my project, my BSc Honours project, where I was trying to study gastrulation and look at the effect of the nucleus and cytoplasm on the unfertilized on the nucleus, and but all the way through, and thinking again about DNA, I was influenced by the wonders of chromosomes. And these two images are taken from Ballinsky's book. And first of all, the first time you could actually really see chromosomes beautifully was in these salivary glands of these insects. And again, here's an induction. These puffs were induced in Chironomus by injecting the larvae with ecdysone, a hormone that allowed this duplication of the DNA, so that ultimately you could see the different chromosomes. But also, this is a picture also taken from the book, and this was the first time I realized that you could see the activity of genes on these chromosomes, because here's the chromosome, and here you've got the RNA being produced on these chromosomes. So not only can you see in certain systems the DNA itself, but you can see the RNA, the activity of the RNA that's being produced off those chromosomes. So I was first introduced to this picture by a wonderful conference that Barry Fabian organized, and this was the figure that was used to advertise, as far as I remember, the conference. And this is taken from a book on the generation of animals by William Harvey, who was actually at Barts and the hospital that I work with. From the egg, everything. And I love this picture because here is exactly what we're trying to do, to understand how you get from the fundamental egg to all of these different organisms here. What is the process by which we end up with these thinking, a human being, and all of these beautiful organisms? One of the other things that I found really inspirational in the book, and that has remained with me, is in the book where, right at the beginning of the books, there is a kind of the science of embryology is discussed. And I've always remembered this little, at least these phrases here, that analytical embryology has to be seen in the light of descriptive embryology. So you need to have really good understanding of the morphology of what you're looking at. You need to compare between different species to look for the universality of any process and of any system. And eventually, you've got to test the causality of the mechanisms by doing experimental embryology. And I guess these three key elements have stayed with me again through everything that I've done during my career. For me, as I said, I focused on chromosomes, and I have taken this one step further, that the motto for my life is from the chromosome everything. And these are metaphase chromosomes that we prepared from normal human blood We have labeled each chromosome 1s, for example, in yellow, chromosome 2s in purple, and so on, so that you can identify individual chromosomes. And so we've been trying to understand how do these chromosomes function, what's the structure of them, how do they function, and in turn, how do they function in health and disease. So what I'm going to tell you about, I tried to get rid of this here, which I don't think I can do, but let me just see if I can move this off. No, I can't. How are genes organized? I'm going to tell you about how are genes organized on chromosomes and the work that we've done in this, in health and disease. Secondly, what defines the three-dimensional architecture of chromosomes? And running through my entire career has been what are the genetic origins of cancer. And here I have that same metaphase spread, but showing you also a G2 nucleus where you can see the chromosomes occupying individual territories here. It's quite fat. It's going to be dividing soon. Here you've got a G1 nucleus. You can also again see the chromosome territories very closely, tightly bundled up together. This is a normal metaphase karyotype. And here is a karyotype from a very aggressive brain tumor called the glioblastoma that I've studied for many years, where you can see huge numbers of genetic abnormalities, where you can see the chromosomes, one chromosome joined to another, multiple copies of some chromosomes, and all of these have been the basis of all these genetic abnormalities in these tumors and others for my work on cancer. Let's first come to how are genes organized in chromosomes, in genes in health and disease. Just to give you 3 examples, my lab mapped hundreds and hundreds of genes to chromosomes using, first of all, radioactive probes, which we would label label and hybridize to metaphase chromosomes. We then progressed onto fluorescently labeled probes, so you could identify the positions on chromosomes. This is, of course, way before the genome browser, which is our Bible that we use all the time in a genomics lab. We also developed techniques for stretching out the DNA. So, for example, this is about a megabase worth of DNA over here, but then you could apply the fluorescent probes over here to get an order of probes. For example, if you labeled the next one in red and yellow to make red to make the red, or if we combine the colors to make green over here. We also worked very closely with cancer predisposition labs, and we were very interested in colorectal cancer and in predisposition syndrome, polyposis coli, where the colon becomes carpeted with little polyps. And we knew that it was a genetic susceptibility. And we found a set of patients who had both mental retardation as well as polyposis coli. And we hypothesized that there would be a chromosome deletion or defect at the site of the gene, which also confirmed susceptibility to polyposis coli. And here you can see one of these patients, actually it was a set of twins. This is the normal chromosome 5. You can see it has 3 lovely bands in the center here, but one of this patient's chromosome 5s only had 2 bands. And this was the first mapping of the APC gene, which is, of course, so critical in development of part of the Winter pathway, to chromosome 5. And this is just an example of how we've used both in situ hybridization techniques, but as well just pure morphological mapping to be able to identify the positions of genes. And of course, then we were part of a consortium to do the exact mapping of the APC gene on chromosome 5. For many years, I tried to study the 3-dimensional architecture of chromosomes, and I used the major histocompatibility complex, the MHC, on chromosome 6 as a model for this. And the reason I did this is because the MHC is constitutively expressed on B cells, dendritic cells, because it's so important in presenting antigens. However, in other cells, such as fibroblasts, you can induce the MHC using interferon gamma. And this is the work that we showed, and it was the first time it was ever shown that when a genomic region is active, you get a loosening of the chromosome and you get a giant looping of the chromosome. So here is a nucleus over here, a labeled chromosome 6 in green over here. These are the two chromosome 6s. You can see it bundled up as a nice territory. This is in a fibroblast where the MHC is silent. These were two probes on chromosome 6 on the MHC. And here, when we induce these cells with interferon gamma, you see within 10 minutes a giant loop coming out and extruding from the chromosome. Of course, this was just a morphological change, but now it's very clear with so-called high-C techniques that gene expression and morphological changes in chromosomes are really directly related to the folding at a gross level, but also at a very, very fine level of the chromosome, of the chromatin. And we then went on to look at CTCF and other markers of chromosome looping and so on to try to define this. And we were able to look at the signaling pathway that led to the extrusion of these loops. And of course, the extrusion of the loops means that the chromatin is in an open configuration and is therefore accessible to transcription factors. Then I'm going to move on to cancer now. So for many years, we studied numerous kinds of cancer. We did chromosome analysis of these. We combined it with fluorescence in situ hybridization. This is just some of the different types of tumors I've published on. We were the first ones to show that the acute promyelocytic leukemia has got a 15-17 chromosome translocation that involves a retinoic acid receptor. And interestingly, retinoic acid is a critical treatment now for this kind of cancer. I've showed many chromosome rearrangements in many other kinds of tumors. This is just one example of ovarian cancer. And in each of these cases, we defined the breakpoints, the critical breakpoints, and tried to see which genes were involved. Glioblastoma, this very aggressive kind of brain tumor. I've done lots of work on that, models of glioblastoma. You can see again the rearrangements that there are here. And then we also worked with clinicians to develop methods so that without getting the chromosomes themselves or even doing molecular analysis, just on a histological section, you could see where you've got clinically relevant genetic changes. For example, this is a breast carcinoma where you've got amplified HER2, and instead of just seeing two tiny dots within each nucleus, we can see multiple copies of this HER2 cell surface receptor, ERB2 receptor, here in each of these. So we could work very quickly with a histopathologist to tell the clinicians whether a tumor had got a poor prognosis or whether it needed to be treated in a particular way. So now moving on, what are the origins of children's cancers? What's really been extraordinary to me is how, over the years, I've realized that certain childhood malignancies reflect abnormal processes of embryonic development. And this is the theme that really has driven all of my work on childhood cancer. We know that there are about 400,000 children who develop cancer globally each year. Of course, you've got huge differences in detection and treatment and so on across the world. But we know it's the leading cause of death in childhood brain tumors. There's a distinct spectrum of the different kinds of brain tumors, different kinds of tumors at different ages. So you might get certain kind of brain tumor at one age range, another kind, say neuroblastoma, slightly higher up, and so on. But there are no consistent environmental factors that have been identified. And this is what I'm going to tell you a little bit about now, that certain childhood malignancies reflect these abnormal processes of embryonic development. And I'm going to give you just 3 examples that I've worked on in the past. This is not from my work itself, but Wilms tumor is a very nasty children's cancer of the kidney that affects the kidney here. It can be hereditary, it can be, in which case it's bilateral. It's actually a silent tumor. a tumor can be quite advanced at the time that it's detected, and often the parent will just detect it by seeing a bump, feeling a bump on the back. But Wilms tumor is really, it has been identified as being a failure of the precursor cells of the kidney to differentiate. And the molecular basis of this involving the WT1 gene working through FGF and other key transcription factors that are downstream from FGF are absolutely critical to this. The same is true for retinoblastoma. Retinoblastoma is a childhood cancer, very prevalent in sub-Saharan Africa. This is from leukophoria from a child with retinoblastoma. You can see the white showing, and this is looking in the eye at a retinoblastoma. And this is caused, most of them are hereditary and involve the RB1 gene, which is important in the cell cycle. And this is, again, it's shown to be a failure of differentiation of the cone cells within the retina. The same is true for neuroblastoma. Neuroblastoma is a malignancy of the sympathetic nervous system. And the sympathetic nervous system, of course, derives from the neural crest. You've got these transcription factors which play a key role in formation of the adrenal glands in the sympathetic nervous system. And it's been found that there's an exact parallel in terms of the genes and the processes between neural crest development and the early stages of neuroblastoma. And essentially what it is a failure of these neural crest cells to differentiate, and they stay cycling in a stem cell-like state. So let's go on to brain tumors now. For the last 20 years, I've been focusing my pediatric work mainly on children's low-grade gliomas. These are the most common primary CNS tumors in children. They are slow-growing and non-malignant, but it's a chronic disease, and even if a child survives, they will have lifelong physical, hormonal, and cognitive impairment. We discovered that most tumors have driver mutations that affect the MAP kinase pathway. And clinical management has been really not really knowing what to do with these, and most of the time it's watch and wait. And because the tumor grows slowly, but then suddenly it can cause a problem. So surgery, radiation, and chemotherapy, but now as a result of the discovery of the MAP kinase alterations, targeted treatments are in development, and I'm going to tell you a little bit about this discovery in particular. Let's look at the MAP kinase signaling pathway. This is extraordinary, this pathway. It takes signals from the outside of the cell, as you know, through the cell surface receptor, and transduces them down this RAS, RAF, MEK, and ERK pathway, phosphorylating as you go along. And this is a key pathway for developing whatever metabolism, whatever process you can think of. MAP kinase seems to be involved in one way or another. There are hundreds of targets of this ERK pathway, of this phosphorylation here. It phosphorylates hundreds of cytoplasmic targets, and the nuclear targets are usually transcription factors. But importantly, the output is determined by the duration and the strength of the signals, whether it's cytoplasmic or nuclear. There are multiple regulators at each tier, which I haven't put in here. There's also pathway crosstalk, which I will show you in a few minutes. We found that we discovered these KIAA BRAF fusions that affect this BRAF gene here in most cases of low-grade gliomas, and a BRAF V600E mutation in a few cases, about 5% of cases. And occasionally, working with my colleagues across the world, we found a few other key mutations, but extraordinary. They all feed into this MAP kinase pathway. So the BRAF is the most commonly mutated gene in pediatric low-grade gliomas. It's a kinase. It's got an activation domain here, a regulatory domain that folds around and regulates this activation domain. This is the fusion that you get from these two genes. It's caused from a duplication here on these genes. removes the regulatory domain, gives you just the kinase domain over here. And the alternative mutation, the BRAF V600E, actually is just a single base pair mutation over here that keeps this constitutively active. And the net effect of these is that the MAP kinase pathway is constitutively on, so you get signaling constantly down. So how does this arise? Well, we know that it arises from a tandem duplication on chromosome 7, that's within this little region that we showed. And it breaks and you get this fusion joining here between these two genes. We then studied about 40 of these tumors that had the fusion. We studied the breakpoints, and we made the most unexpected discovery, and that is that we get microhomology, tiny little regions of homology on the two genes which fuse. So this is, for example, the gene KIAA gene, and this is the BRAF gene over here, the sequence. And we found this little region of identical homology. Most of them had homology which were exactly at the breakpoint. We then deduced that this must be arising through a mechanism of microhomology-mediated break-induced replication. It's something that had already been described. And essentially that during the process of replication, you get a break, a nick at one end, at one part of one of the strands. This then loops around, it becomes dislodged, it loops around, and on the basis of microhomology within a sequence, in this case, at a different position on the chromosome, leads to a re-replication of this region here. And that has also been confirmed in other tumors by others. So let's finally come back to this pathway. This is the deregulated pathway in low-grade gliomas. We find activated pathway, it's on conserving the tumor, but we propose that the genetic abnormalities not only change the dynamic behavior of this particular pathway, but rather change, because this pathway is actually linked to lots of other pathways, for example, PI3 kinase pathway over here, and there are other pathways which would feed into this as well. We hypothesized that the genetic abnormalities change the dynamic behavior of the entire signaling network, and they disrupt feedback and crosstalk to and from other pathways. This leads to aberrant input and output relationships and dynamic changes that drive tumorigenesis and alter the phenotype of the cell. And that importantly, targeted inhibition of this pathway affects the whole signaling network and must be understood in this broader context. So we generated multiple multi-omic sets of low-grade gliomas. We've characterized the signaling and regulatory mechanisms, and we've just started to look at the response of these tumor cells to targeted inhibitors. I'm very quickly going to go through these. These are two of the common types of tumors. These are the most common low-grade gliomas over here, usually in the cerebellum. We took glioneuronal tumors here as well, plus normal and controlled brain tissue. We did analyze the proteomics and RNA coding and non-coding RNA, total proteomics, phosphoproteomics, and so on. And we've put all this together into an integrated network analysis. And instead of these are two images from the two different kinds of tumors. Let's just focus on this one here, on the pilocytic astrocytomas. These are the most common tumors in childhood. They appear in the cerebellum. These are the signaling pathways that we find are altered in these tumors compared to the normal controls. And just to show you, this is the MAP, RAS, RAF, MEK, ERK pathway that I showed you earlier, but we consistently find that AKT is also involved, that we get NF-kappa B, of course, involved, it feeds off here, and that this is part of a much broader pathway that we're deducing from the enriched kinase activity that we're seeing from our phosphoproteomics in each of these tumor types. And we see the same in these glioneuronal tumors. These tumors have got a V600E mutation. These have a BRAF, just the BRAF fusion. We've done similar studies we have also looked I won't go into this into detail, transcription factor enrichment analysis, trying to link the different kinases with transcription factors and their targets which are involved, and we're then now able to put these together into trajectories that allow us to see, for example, the expression of particular cell surface receptors, of particular proteins that we know are important in cell growth and in the phenotype of these cells. And finally, what we're doing, because we want to eventually link this in ultimately, because it's very hard to grow these tumors because they are slow growing in culture, so it's very hard to get to the fact there are no good tumor models. So working with my colleagues at the Dana-Farber Cancer Institute at Harvard, we are studying isogenic models made with neural stem cells that contain each of these different abnormalities, and we are now conducting again this large study over here, multi-omic study, and this is going to allow us to identify the network analysis that give us the network changes from each of these different abnormalities. But most important is going to give us a means by which we can experimentally test for causality and also test with drug perturbations to see which are the best drugs to use, for example, to slow down the growth of these tumors. So I've whizzed through this very, very quickly. In conclusion, just to the brain tumor part, we've identified regulatory signaling networks that have been identified in children's low-grade gliomas, and they reflect the rewiring of critical pathways in these tumors arising from the major genetic alterations. And our current studies are looking at the responses to targeted inhibitors. I'm sorry I've spoken very quickly. I'd just like to end with, actually, which is one of my favorite images that I think hasn't been shown yet. This shows Professor Ballinsky, and I think it really shows so nicely the atmosphere that there was in his department. It was a very kind atmosphere. He loved what he did. You can see how this hand, even to this hand on here, holding Professor Nolte, somebody, I think Mrs. Heslop, giving a present to Professor Nolte when he retired. This was the nurturing and purposeful and engaging and the most amazing department that I was so fortunate to work on. So thank you posthumously to Professor Ballinsky. I'd also like to thank Professor Pillay and Professor Barry Fabian, and the organizing committee for the symposium for inviting me to speak here. And of course, all my scientific collaborators who've helped us, and our funders, most importantly, the patients and their families. So thank you very much. I think I can stop my sharing here. Stop share. There you go. Hope that was okay. Went across well.
Thanks, Denise. This is Neville Pillay again. I'm afraid you lost most of us along the pathways because it's really a complex talk. I've got one question for you, and maybe this is showing my ignorance. I mean, I've done chromosomal work many years ago, really looking at fusion chromosomes and how that creates speciation events. So it's unrelated to what you're talking about. But these chromosomal mutations, you talked about alignment of certain segments that cause these anomalies. Do they arrive spontaneously, or is there certain predictors of those?
Yes.
Anyway, that's my question.
So you've got this microhomology, of course, from the sequence right across the genomes. And it just so happens that during replication, where you've got a break within one of the segments of DNA, because you've got microhomology and because the DNA is folded, this is going to come around and bind, hybridize to a region, it could be on another chromosome, but in this particular case, it's to a region that's higher up on the chromosome. And actually, this has been proposed as a mechanism for generating genomic alterations that give rise in time to speciation. So it has been described elsewhere. This is a phenomenon that has been well described in other systems. So we were quite amazed to see this microhomology at very high statistical significance in these genomic regions. Hope that answers your question.
No, it does. And it does put my early studies into perspective too. But obviously, not only speciation, but other diseases emerge from these sorts of homologies. I don't know if there's any questions from anyone else.
There is a question online.
There is an online question. Denise, I would like to ask you, people have done a lot of work on transcription factories. I wonder what kind of changes you may see in cancers in your models.
We haven't looked specifically at transcription factories, but I love the idea of transcription factories. And when we were doing all our three-dimensional studies of chromosomes and the looping out, actually we did find that there are certain organelles, for example, PML bodies, into which some of these looped into. So I think that the technology has advanced so much now where you're able to look at the chromosome folding using this high C technology. But I think as microscopic techniques have improved and fluorescence images have improved, so you get much better resolution, I think this is the perfect time to start looking again at those transcription factories, which I know exactly. I was very excited about transcription factories. And also we used to find splicing factors with the transcription factories. So we'd look at a nucleus, we'd have the chromosomes, but you could also see the transcription, so-called transcription factories there with high concentrations of splicing factors. If I had enough time, I would love to do this and continue to do this. But thank you. That's a lovely memory for me.
Thank you. Are there any more questions online, maybe? No, I think that's the... Sure.
Neville, may I have a question?
Yeah, let's go ahead, Alex.
Thank you, Dennis, for this very nice presentation, and we can see how the scientific school of Boris Ballinsky develops. And I have a more precise question. Is there any contribution of pseudogenes, I mean mitochondrial copies in the nucleus, to the development of different types of cancers? So contribution of pseudogenes to cancer, is there any?
Sorry, what kind of genes? Mitochondrial?
Yeah, clear DNA. Yeah, well, this is the problem what we have in barcoding, for example, when mitochondrial copies are present within the DNA, which is in nucleus. So my question was, is there any examples of such pseudogenes?
Pseudogenes.
No. Pseudogenes, yeah. Is there a contribution to cancer?
Not that I know of. Actually, the big area, it's maybe not really exactly the same that we're starting to look at now, are transposable elements. And just thinking about something that's not the conventional DNA or conventional genes. And actually, we're looking at the different tumor types. We're looking at expression of transposable genes, and we find distinctive expression of these genes, which must have some effect in the different tumor types and in our different controls. So I think the better our molecular biology gets, our ability to profile, I think also using AI, I think we will, as it is, even with our signaling networks, bringing in microRNAs, non-coding RNAs, long non-coding RNAs. We're finding it quite difficult to keep tabs on everything. And, how does one begin to study it? So I've approached the transposable elements with some wariness. But this, as far as pseudogenes or mitochondrial genes, I'm not aware of anything like that. Okay, but I will look it up.
Thank you.
Let me ask again, is there any questions online? Denise, I can't see the Zoom link, so I can't see if there's any hands.
There is one hand up.
Okay. Olesi.
Hello. It was very interesting for me. And in the last slide, I saw long non-coding RNAs. And can you tell just a brief? So is it an evidence they are doing something, and what?
Oh yeah, we've done so much profiling with long non-coding RNAs. We have got a really good batch of long non-coding RNAs that are highly expressed in one tumor type compared to another. The trouble is we have no idea what they do. There are a few long non-coding RNAs. One of my colleagues at Bart's, Stojic, Levorka Stojic, is looking at long non-coding RNAs which are important in cell cycle, and we're working with her on those. But we find some long non-coding RNAs that are 200 times more expressed than we expect in one or other of the tumor types. But the trouble is, it just occurs, it's near, in this case, near an NTRK gene, a cell surface receptor gene, that itself is fairly highly expressed. What's the connection? We just don't know. Is it an enhancer? We just don't know at this stage.
Maybe expression noise. No?
It's expression. It's expressed. So,
but maybe it's the noise of expression.
It may just be. It may just be. Perhaps I will speak to you offline, and maybe you can help us to try to work out the best strategy to try to find the significance of these. Thank you for the question.
Thank you.
Denise, we out of time, but thanks very much for making yourself available. Again, a very interesting talk. Trying to explain all these complexities and something that's very close to all of us in terms of cancer and so on. So I'd like to wish you a very good afternoon, and take care. And the symposium was much better for having you present. Thank you very much.
Thank you.
Speaker is Professor Vladimir Kosh. He's visiting professor at the International Institute of Molecular and Cell Biology in Poland. And Vladimir was one of the first people that we got on board in terms of the symposium so long ago. And Vladimir, you're going to talk about a century of research on the primordium, limb induction to K channels in development. I didn't know whether K referred to potassium, but I left it at saying K for now. Over to you, Vladimir, and thank you very much for your participation in the conference.
Thank you for the invitation. K is indeed potassium, so we're going to continue from that. Yeah. Can you hear me?
Yes, we can.
Okay, super. So, as you already know, Boris Ballinsky graduated from Kiev University when it was still Saint Vladimir University, whereas when I graduated from that, it was called Kiev State University. Now it is called Shevchenko University, National University of Ukraine, and so on. So you see, names have changed over the years, and when Boris retired, I was still a student at this university, and at the Department of Zoology, there was Professor Kistyakovsky, whose brother apparently was also a refugee, and he became famous for working in Manhattan Project and later on being scientific advisor to President Eisenhower. So there was a bit of a history in this department, and people were often speaking in half tones, in sort of trying not to tell too much in those days. And one of the things which they didn't want to talk much was Boris Ballinsky. So obviously moving to Wits was a significant improvement for him. Otherwise, probably he would face grim future in Ukraine. Anyways, so because the enemy ears were listening, we were all sort of hush-hush. So I became interested in this name and started to study the history of Boris Ballinsky, which eventually led to publication of several papers in scientific journals. And one was published quite recently, end of last year, where his name is again mentioned. Okay, so everybody's showing this book or copies of other editions. What I want to bring your attention to is this ex libris, or the sign which Boris used for a long time in his library and marked his books. So this five-legged creature was created by him in 1924, somewhere. In paper was published in 1925. So obviously, if you look at the history of Boris's publications, you will find out that he was interested in development of limbs for about 50 years. And the first publication in 1925, the last publication on transplantation of ear primordium was in 1957, and even was another one where in formation of supernumerary limbs in Anura was studied by Boris Ballinsky. So it shows great interest of his in this particular subject. So again, several speakers demonstrated these images from his early publications. What I want to bring to your attention is this particular book, which was published in Russian in 1936. And I suppose that this book is probably from the library of Ivan Schmalhausen, mentor of Boris Ballinsky, because it was given to me via Sergei Vasetsky from Olga Schmalhausen, daughter of Ivan Schmalhausen. Well, anyway, this book is here. You can see it's really exists, not only in PDF. And I want to say just a couple of words about Sergei Vasetsky, because he was quite active as a translator of English literature. And while traveling to international conferences, he met with Boris and asked whether Boris would like his book to be translated in Russian. And was then Boris gave him several copies of this book. So one of these copies ended at the Department of Embryology in Moscow University and was used to teach students in absence of other complete sort of set of texts on developmental biology. Whereas when Sergei sort of contacted publishing house, which was involved in publication of translated literature, he was told in no uncertain terms Ballinsky book cannot be published in the USSR. He was prohibited or forbidden. Anyways, so coming then to experiment, interesting as to what kind of events taking place during these processes. FGF speakers during this conference. So if you look at expression of some of these FGF genes, there are about 20 of them, but a couple of those I expressed quite early, and you see here pictures of expression of FGF3 in the otic vesicle of zebrafish in this case, and then expression in bronchial arches. Okay, and another gene, FGF8, is also expressed in the ear more continuously. So people were wondering whether FGF signaling may play a role in inducing limb buds during transplantations. So these experiments with transplantations were repeated in different model. So in chick there is a possibility to transplant either primordium of the limb or bead in this case, and such bead can be implanted in the same position. Such expression of the bead where in these embryos where these experiments were performed. So in essence, it sort of answered the question as to what were the developmental factors involved in induction of the limb, at least those which were coming from the otic vesicle. But one need to remember also that otic vesicle expresses TBX genes. It expresses TBX2 and TBX3 genes. TBX genes are members of the family which was originally discovered when the brachyury gene was found in mammals in 1927, actually by another Ukrainian refugee, Nadezhda Dobrovolskaya-Zavadskaya in Paris, who generated the first radiation mutants in mice. Anyway, so related genes were found in early 1990s, the whole set of these genes, and it was found that in the eye there are a couple of these genes expressed. These are transcription factors interacting with DNA directly. And there is also a couple of these genes which are expressed in the lateral mesoderm. So in position of TBX5, the anterior limb is forming, and in position of expression of TBX4, the posterior limb is forming. So when the bead is transplanted into the flank between these two zones, so the sort of hybrid limb is forming carrying features of anterior and posterior limbs at the same time. So it seems that this evolutionary pathway is quite conserved, involving TBX genes, FGF signaling and TBX genes interactions in the otic vesicle, and again, other TBX genes and FGF in the limb. Okay, so by the time I spent quite a bit of time doing research in Southeast Asia, in Singapore, where actually my first papers on Boris were written. But by the time I sort of finished my presence in there, I already developed interest in formation of hollow structures, such as brain ventricular system and in the ear during development of zebrafish. So we obtained a couple of mutants which were shown formation of hydrocephaly or demonstrated the role during formation of the brain ventricular system. But it also was found that they also play similar role during development of in the ear. So here you see the schematic of the early otic vesicle with various structures such as semicircular canals and sensory patches forming here in this very tidy structure. And organization of the inner ear of zebrafish is similar, significantly similar to that in human or in mammals in general. So they also have sort of sensory cells with long kinocilia and with stereocilia, which detect sound, etc., etc. There are also some progenitor cells nearby. Okay, so we got these couple of mutants, and when we sequenced and identified the mutation, we found the mutation were in the subunits of potassium channel. So here you have a schematic of the tetramer of one of the potassium channels, which is known as KCNB1. This is electrically active alpha subunit of the voltage-gated potassium channel. And they play important role in establishing the resting potential in cells, but also during nervous impulse. But at this point, there are no such events happening as yet, and expression of these genes take place largely in the cells lining the hollow organs, so they are not in the excitatory cells. Mutations of KCNB1, which is also known as KV21, that comes from electrophysiology studies, is known to cause in human epileptic encephalopathy. So the structure of this molecule is such that it contains two long cytoplasmic domains and 6 transmembrane domains with electrically active pore, which form these complexes. And these complexes can be formed from only electrically active, which on their own have no activity as such. So you can see expression of this electrically active subunit. It's expressed in brain ventricles, but also it's expressed in the cavity of the otic vesicle. And this is in high magnification. You see this sort of darker regions where this gene is expressed. So we got one of the transgenic lines which nicely defines development of the otic vesicle, and we use these for observing development under the microscope, so-called in vivo. So you could see the otic vesicle. You can see this semicircular canals formed in optic bright field otoliths, which are sensory stones of the zebrafish ear. Similar structures exist also in mammalian ear. But in a mutant, this cavity is much smaller. Some of the elements in the serialization of otic vesicle of otoliths is abnormal. But plus, of course, we performed behavioral experiments demonstrating that these animals are abnormal in balance. So here we have sort of quantification of all these processes of which I was talking, area of the otic vesicle, size of the otoliths, two different kinds of those at different developmental stages and so on, demonstrating that indeed such difference exists. Okay, so why these vesicles are smaller? Well, it could be due to number of reasons, one of which could be insufficient sort of really generated by potassium channels, and but also in addition we detected that cell proliferation is much reduced in this particular, in the mutant affecting electrically active subunit. And here is quantification of these measurements. So another element of the inner ear affected is kinocilium. Well, I must again emphasize that so these are done using experiments microscopy was done using high resolution light sheet microscopy. Here is organization of the kinocilium in wild type animals, and you can see that they sort of normally quite perpendicular to the surface of the in the ear, they extend into the cavity quite straight and so on. But in the mutant, their organization is affected. They are short and they are not developing in a particular direction. So here is quantification of this process. So, and it's not because of the absence of cells which may generate kinocilium. The cells themselves are present, seems to be present, but the kinocilium here, which is present in wild type, does not develop in the mutant animal. So most likely it is deficiency of kinocilium, not of the cell itself, although the number of these cells could be lower. Anyway, so in summary, we described the mutant, which affects electrically active subunit of potassium channel. It's so-called loss of function mutant. It results in reduction of the ear and otoliths, defects in cilia, and abnormal balance of the animal. Okay, so I mentioned previously that there are two types of subunits, one of which is electrically active subunit, and another one is silent subunit, so-called, or modulatory wild type, which is usually compared to electrically active subunit, have much shorter C-terminal cytoplasm. Several mutants, two of which are shown on this schematic. So in one case, we have truncation of the C-terminal region, and in another one, mutation causes change of the open reading frame. So it results in the formation of an ectopic region of stretch of amino acids, which are which creates domain end of the protein out of the cell. So instead of cytoplasmic, this C-terminal region becomes extracellular. And due to presence of hydrophobic residues, amino acid residues. And if you do electrophysiological measurements, so in this case, one can see that on its own, electrically active subunit is relatively highly active. In presence of the wild-type subunit of silent subunit, activity will be much reduced. So it is acting as a modulator of electrical activity of the channel. Then in the C1 mutant, that is this one, you have slight loss of function for modulatory subunit, so therefore channel becomes more active. Whereas this particular mutation results in almost complete shutdown of the channel activity. And there was another mutant previously described. Okay, so if we look at expression of modulatory subunit at RNA level, here in situ hybridization demonstrates presence of this transcripts inside the otic vesicle. And when we look at development of left and right otoliths in wild type forming in each of the otic vesicles, in the somewhat increased size of the otic vesicle, and in some of the otoliths becomes smaller comparing to that in the wild type. And this process can be confirmed by analysis of genes which are expressed in the ear. So, for example, those FGF3, which I already mentioned, or TBX genes, expression of these genes is deregulated from increase in transcription to reduction of this process in respect of different genes. But C1 mutant is, or C2 mutant is of particular interest because it has such a striking phenotype. Whereas otic vesicle forms rather normally, which is quite surprising for such severity of the mutation, the otoliths are completely abandoned and never developed in these mutants. So molecular structural analysis demonstrates that these ectopic or additional C-terminal transmembrane domains are sort of forming the complex with other domains and most likely interfere with their activity. So instead of cytoplasmic orientation, which would be somewhere around this area, they are in the membrane and probably messing up the ion penetration through the membrane. Okay, so I was not able to put all the data. Of course, I just show you snippets of several papers, but trying to combine history and modern type of research, we see that experiments which were started by Boris Ballinsky a long time ago, almost 100 years ago, were not only led to successful resolution of the molecular mechanisms, demonstrating that it is an interaction between FGF signaling and TBX genes which take place during formation of ectopic limb, but also it sort of continues in a number of different ways, including analysis of physical factors such as bioelectricity. And I've just shown to you two mutations into different subunits of potassium channels, which in the ear and also in other organs such as brain ventricular system and others which we're still working on, have antagonizing effects, and mutants, and these mutants have opposite developmental phenotypes, which are quite informative and useful when experiments are planned and experimentator wants to be certain that he will obtain effect which he expects to see. Although in certain situations, such surprises like those seen in the PCNG4B-C2 mutant are, of course, welcomed also. Okay, thank you very much.
Thank you, Professor Korsch. Are there any questions online? Let's do online first before we go to the audience here. Is anyone online?
Yeah, there is a question from Bob.
Dear Vladimir, it was very pleasant for me to hear your lecture, and I just wanted to ask this question. That there is a movement to merge electrophysiology and development biology. There is Michael Levin in Boston who is doing for a lot of time, and this group, they believe that elevating of expression of potassium channels leads cells to more proliferating stage. Can you say something about this?
Well, it's a rather complex question, and I do not want to be sort of single-minded in this respect, because it depends, like many things in biology. So, for example, apparently effects of deficiency or gain of function of potassium channels will be somewhat different depending on the type of cells that we are looking at. So in adult cells, this situation is different from the one which happens in proliferating cells or stem cells. So actually it could be quite opposite. So it all depends on the context which is discussed. Okay, so having said that, my own results, for which I'm responsible for, demonstrate that in KCN-B1 mutant, which essentially loss of function of electrical activity of potassium channel, we observe reduction of cell proliferation. Okay?
Thank you.
In the mutant KCN-G4B-C1 or N-terminal truncation of the channel, which is also a loss of function of the modulatory subunit, which results in increase in activity of the channel. We observe increased cell proliferation. So in development, it works. This ability of potassium channel results lower level reduction of cell proliferation.
Thank you very much.
Thank you. You were breaking up a bit, Professor Korsch, but we did get your answer. The audio came through. Are there any other questions online? There are no questions from the audience here, so I would like to thank you again for making yourself available and presenting this afternoon. Again, a very interesting talk, building up on the early work from Professor Ballinsky. And do have a good afternoon, and we'll hopefully meet again in the future. Thank you very much. Our next speaker is Dr. Anna. Dr. Shevchukova. Can you just give me a second? I'm just introducing you. Nice to have you, can hear you online. So Dr. Hanna Shevchukova is from the Schmalhausen Institute of Zoology. I think we heard someone this morning speak from that institute. And you're going to tell us about variation of the chronocranium development in snakes. So over to you, Anna.
Good afternoon. I hope you can hear me good. Tell me something.
We can hear you.
Yeah?
Yes.
Okay.
We can hear you nicely.
Okay. Dear colleagues, I'm very glad to be here today and to present my report. And first of all, I would like to apologize if during my report you hear non-English words. It is because my language mixing, but I'll try to do my best. So I work in the Department of Evolutionary Morphology. The department of classes of vertebrates were continued in our department. These are mainly comparative and descriptive study of the development of the skull and limb.
Recording in progress.
And one of the first work that I read was the work by John Pringle, who worked in Natal Museum in Pietermaritzburg. And this is the first work to compare chondrocraniums in different species of African snakes. The author paid great attention to the differences in the middle region of the chondrocranium, the presence or absence of carotid foramina throughout carotid arteries passing and fenestra X. And Pringle paid great attention to this region in snake's chondrocranium. Such differences aroused my interest in whether the cartilaginous skull of one group of animals would really be so different. Of course, there is nothing surprising here, because the differences in morphological characters are the norm in different species. Inferences about systematics and evolution are based on their presence. Later, my supervisor pushed me to study variation in skull morphology, and there was a question. Is the chondrocranium different in embryos of the same species at the same developmental stages? And I did such investigation studying grass snakes embryos. To study the variation of embryos of the same species at different developmental stages, we simultaneously selected several embryos from the same clutch on different days of incubation. All embryos were removed at the same time, were morphologically identical without external pathologies, and were at the same stage of development. Thus, we were able to obtain samples consisting of 6 to 12 embryos at different developmental stages. And at all, 114 embryos at successive developmental stages were examined. Cleared and double-stained specimens were prepared after the stain technology by Siemens and Van Horn. As a result, the cartilaginous structures of skull are stained in blue, and bony structures are stained with red. And now I will directly present the result of this research. Variation in the formation of cristacellaris has been detected. Here you can see the structure about which we're speaking today. And this is one of the basic structures in chondrocranium of all vertebrates, and it appears between basic cranial structures, trabecula cranii and paracardalis, and divides the space between them into two fenestra, pituitary fenestra and basicranial fenestra. In further development, this cartilaginous structure is replaced by bone, which is called basis sphenoid. And now I'll briefly discuss the process of formation of this structure in grass snake embryogenesis. In this way, we can better understand the further results. So the medial processes from polar cartilage arise and forming carotid incusura, where in future carotid arteries pass. Down this incusura enclosed and in such a way forming carotid foramina. And later, between them appears crossbar, which is called cristacellaris, about which we are speaking today. And now we come back to our embryo samples. At developmental stage 31, we identified one embryo out of 8 in which the carotid foramina were formed, but there was no cristacellaris. All another 8 embryos had fully formed, normal developed structure. We associated this with the probability of a delay in development within the same clutch. However, the results which were obtained later surprised us. In embryo samples at developmental stage 31 and 32 and 32 plus, there were several embryos in which cristacellaris was also absent. On this slide, you can see 3 variants of the formation of the identical area of chondrocranium. Noteworthy is the single variant in which cristacellaris is halfway present. And this process is formed only on one side. And the question arises. Will Christus Stellaris appear at later developmental stages? If not, how it will affect basisphenoid formation? And this study shows that in some embryos, Christus Stellaris does not appear at all. The photo clearly shows here, the periosteum. This is a border of former cartilaginous structure, and here periosteum is formed and ossification begins. However, absence of Christus Stellaris does not prevent the formation of bone, which is called basisphenoid. Depending on the presence or absence of Christus Stellaris, we observed in grass snake embryos two ways of basisphenoid formation. The first one, if the Christus Stellaris is present, here on picture there, if it presents, then basisphenoid is formed totally as a replacement bone, as a bone of chondral origin. And if Christus Stellaris was not formed, as on this photo, then processes of dermal origin appear from both sides, and they grow towards each other and merge. Later they merge. In such a way, the absence of cartilaginous structure is compensated by dermal ossification in embryogenesis. In this way, in the lateral parts of future basisphenoid are of chondral origin, and the middle part of the bone is of dermal origin, and the bone as a whole is of mixed origin. The next slide clearly shows several development variants after the closure of the carotid foramina. When the cartilaginous processes are not formed at all, when it forms only on one side, asymmetry of development, when they formed but not merged, and normal variant when Christus Stellaris is normally and fully formed. And what is interesting that in this sample of embryos, only 4 out of 9 embryos had fully formed Christus Stellaris. In this table, you can see our results with number of embryos which had fully formed normally developed structure and number of embryos in every clutch which had no it. And overall, the absence of Christus Stellaris occurred in 15% of embryos. Thus, in grass snakes embryos, there are 3 variants of Christus Stellaris formation. It's complete absence, it's asymmetric, not fully formation, and fully formed formation of Christus Stellaris. Moreover, the presence of all these variants can be formed in embryos without one clutch. It I would also like to mention here some very interesting and rare cases of atypical description of the formation of analog structure in chondrocranium of lizards. In lizards, analog structure is called acrochordal cartilage and situated in this area. And as a rule, in embryogenesis, it appears as independent cartilaginous structure. And here you can see that the beer described that acrochordal cartilage in Lacerta agilis is formed as a result of the fusion of two processes which later fused. Such a situation is atypical for lizards, and for a long time the beer's description was criticized and assumed to be wrong. In this case, my attention was drawn to the work of the cranial development of Andean lizard. At the one early stage, the authors observed fully formed acrochordal cartilage. At later stages, they observed as they said, resorption of the middle part of the structure, and in later embryo they saw again fully formed normal situation in the lizard's chondrocranium. In this regard, I dare to suggest that the formation of the corresponding area of chondrocranium in lizards, as in snakes, is also variable. The next slide presents my conclusions. Allow me not to read it out since this was mentioned during my report. The problems faced by embryologists, morphologists, as a result of the variation of the studying objects are also presented. And I want to say that variation in the formation of morphological structures is a great problem for researchers who work in comparative morphology and evolution. And additional difficulties arise from interspecific comparison and determination of morphological homology. Thank you for your attention.
Are there any comments or questions for Hannah?
Yes, thank you so much for that presentation. It was really interesting. I was just wondering if the different variants that you found were geographically isolated from one another, or if all of them came from the same population and it's just individual differentiation in how it develops.
All embryos were collected in one place, so it's one population, and it's embryos from one female.
All right, thank you so much.
Any other questions for Anna? Anna, thank you very much. We at the end of this session, but thanks again for making yourself available and presenting on an interesting case of the complexity of embryology, morphology, and those important areas of research that are not easily understood or accepted. But I think your talk showed that. Thank you.
Thank you.
a 15-minute break. There's tea and coffee outside. speakers of the day. The next speaker is Professor Robin Hettam, who was a member of our staff and has now emigrated to New Zealand. Robin has pre-recorded a presentation on phenotypic flexibility of long-lived mammals in a hotter and drier world. We will now play the recorded presentation.
Professor Ballinsky was particularly interested in the loose connection between genotype and phenotype. For more than a decade after his official retirement from the University of the Witwatersrand, he investigated inheritance of wing coloration in these African garden butterfly in a breeding shed he built in his backyard. He was fascinated by how the genes, the genotype, of these garden butterfly could be so varied in their expression of wing coloration, their phenotype. Subtle differences in the environmental conditions to which the butterfly larvae were exposed may have initiated different physiological and behavioral pathways that influenced wing coloration. Professor Ballinsky referred to this as a concealed pool of genetic variability that may be made use of under changed environmental circumstances. Environmental circumstances have certainly changed since then. Hashtag Show Your Stripes has become a popular way to visualize recent changes in global temperature. This figure shows those changes in global temperature from 1850 to 2022. The pale-colored bars shows years of average temperature. The blue bars shows years that were cooler than average, and the red bars show years that were warmer than average, with progressively darker shades of red showing particularly warm years. It's obvious to see how many of those hot years have occurred in the recent decades, with 2023 being the warmest year on record. Africa is particularly vulnerable to these changes in temperature. Not only do we have a low adaptive capacity, but our surface temperatures are increasing twice as fast as the rest of the globe. By the end of the century, many regions are predicted to show an increase in temperature of up to 4 degrees centigrade. But that thermal threat is exceeded by the reduction in rainfall. Many of our arid and semi-arid ecosystems may experience up to an 80% reduction in rainfall by the end of the century, combined with increases in both the frequency and intensity of droughts. Animals' first response to these changing climates may be to shift their ranges, to track that so-called bioclimatic envelope. But that's not always a feasible solution. This scimitar-horned oryx, for example, would need to shift from its historic distribution in the Sahel, and it would then need to move thousands of kilometers to the Kalahari, where it's projected to gain suitable habitat in the future. Not only is that distance likely to be unattainable, but it would have to move through a modern human-dominated landscape in which fences and cities block its way. These bioclimatic envelope models project that if movement is limited, nearly 2/3 of African mammals may be extinct within the next 30 years. Sadly, the scimitar-horned oryx is now extinct in the wild because of a multitude of confounding pressures. If animals are forced to stay put, the only way to survive is to be able to adapt to the changing climatic conditions. But those adaptation options may be limited in many of Africa's charismatic, long-lived mammals. There are a number of individuals that are alive today that will be around to absorb those changes in climate projected for 20 to 50 years from now. Some individuals, like these African African elephant may be around to observe and experience those changes in climate projected for the end of the century. If they are to survive, they cannot rely on genetic adaptation or microevolution. Their lifespans are simply too long. They're going to have to rely on phenotypic plasticity to adapt their physiology and behavior to these changing climatic conditions. Our understanding of adaptation to climate change is still very much at the same point it was over 20 years ago, when just prior to Ballinsky's passing, Holt in 1990 noted that there is almost no species for which we know enough relevant ecology, physiology and genetics to predict its response to climate change. Recent advances in biological technology have enhanced our so-called physiology toolbox to allow better insight into animals' responses to changing environments. While historic laboratory studies have provided valuable insight into what animals can do, we really need to monitor the physiology and behavior of free-living animals in the field, going about their daily business, to assess how animals are responding to changes in environmental conditions when exposed to a multitude of environmental stresses in a natural setting. This figure, from a recent paper by Lucy Hawkes, highlights how recent technological advances now allow us to make a multitude of physiological measurements in free-living animals, what she termed physiologging. For example, we can now measure the partial pressures of oxygen to infer tissue function of diving mammals. We can measure ECGs and EEGs remotely to infer the organ function of migrating birds. Ultimately, we can monitor how physiological systems and free-living organisms are functioning. In the future, we may also be able to link these physiological functions to molecular drivers to differentiate genotypic and phenotypic plasticity. To date, my research has primarily focused on biologging or physiologging of body temperature, which provides insights into the thermoregulation and other interlinked physiological systems to ultimately infer organism functioning while the organism is free-living in its natural environment. This figure shows an example of that body temperature data collected via physiologging or biologging of 6 species of African mammals that were free-living in their natural habitat. We can see that there are some differences in body temperature. Something like the large African elephant generally have slightly lower body temperatures, whereas the antelope or ruminants generally have slightly higher body temperatures. But in general, the rhythm of body temperature, or the fluctuation across a 24-hour day, was remarkably well conserved, irrespective of body size, diet. For example, we've got herbivores and carnivores, or even time of day when the animal was active. Here we've got the nocturnal lion compared to a diurnal cheetah. What we find is that rhythm of body temperature generally reaches a minimum shortly after sunrise, increasing throughout the day to reach a maximum around sunset. And the amplitude, or the difference between the minimum and the maximum, is generally quite small, within 1 degree centigrade. And this is very similar to our own body temperature rhythms. To investigate conditions under which body temperatures may vary, one of my first PhD projects set out to investigate the physiological consequences of color variation in a free-ranging herd of springbok. Much like Professor Ballinsky's butterflies, color variation occurs naturally in springbok, but these color morphs have been artificially increased through selective breeding by game ranchers. First, we wanted to look whether the heat load was different between these different color morphs, and, as predicted, the darker and common color morphs displayed a much higher heat load from radiation than their whiter counterparts. Next, we wanted to test if this then had an effect on their body temperature rhythms. And as you can see, the darker color morphs displayed much higher maximum body temperatures on hot days. And this often restricted the times of their diurnal foraging activity. Conversely, on cold days, it was the white springbuck that struggled to maintain their body temperature. Lower body temperatures in the white springbuck on cold days likely reflected an energy deficit associated with the increased thermoregulatory costs in the cool, dry season. One species of long-lived mammals that may be particularly vulnerable to a hotter and drier African future is the elusive aardvark. Aardvark are a nocturnal, burrowing species, and it has a wide distribution throughout much of Africa, but it is notably absent from the arid desert regions. We therefore used biologging technology to remotely and continuously monitor body temperature and activity of free-living aardvark in the Kalahari, a semi-arid region on the edge of the aardvark's distribution range. These two plots then show the 8-month body temperature rhythm of 2 of our aardvark. You can see that the body temperature generally ranged between about 35 and 37 degrees centigrade on a daily basis. And the aardvark were able to maintain this narrow rhythm of body temperature throughout that 8-month period, despite much larger fluctuations in ambient temperature, with air temperatures exceeding 40 degrees in the hot Kalahari summer and dropping below freezing in winter. However, during a hot and dry summer drought, aardvark started to show a decrease in that rhythm of body temperatures, minimum body temperatures occasionally dropping to 30 degrees centigrade. These orange arrows represent the time when the aardvark died. 5 of our study aardvark died during this extreme summer drought, and we found the remains of many other aardvark at the same time. One of our implanted individuals did survive the summer drought. And this figure shows the body temperature of the surviving individual, the corresponding air temperatures, showing by minimum, mean, and maximum air temperatures, and the vegetation productivity, highlighted by the green line, together with the rainfall that was prevalent on site. You can see that during the drought, the rainfall was much lower than the same period during typical years. And even our surviving aardvark showed these low minimum body temperatures during the drought. We then implanted additional animals during the dry winter months to try and get a better understanding on what was causing these fluctuations in body temperature and what had caused the death of so many of our aardvark. This figure then shows the body temperature rhythm of one of those new aardvark over a 2-year period. And all of our implanted aardvark showed these extreme reductions in minimum body temperature in the dry winter months following the summer drought. When rainfall returned, body temperature rhythm narrowed, and it remained quite narrow despite fluctuations in ambient temperatures and some fluctuations in vegetation greenness throughout the subsequent 2-year study period. With the aid of camera trapping and activity biologgers, we were then able to determine the time of emergence, represented by these green dots, as well as the time of return, represented by purple dots, to the burrows.
The lower line shows the time of sunset. The upper line shows the time of sunrise, so that all points highlighted between these two lines represent the nighttime period. During typical years, aardvark would generally emerge just after sunset, returning to their burrows around sunrise, highlighting their typical nocturnal activity rhythm. During typical winter months, these dry winter months, there would be a slight shift in that activity pattern, with aardvark emerging in the late afternoon and returning to their burrows in the early morning. But during the summer drought, aardvark shifted to an almost completely diurnal rhythm of activity. Our aardvark were emerging around midday and returning to their burrows just after sunset, with similar patterns evident in the subsequent dry winter months. So what was causing our aardvark to then have these low minimum body temperatures, despite being active during the heat of the day? We think that it may have been related to their prey. Aardvark rely almost exclusively on ants and termites, and our aardvark had a preference for harvester termites, with 82% of their energy requirements and 89% of their water requirements met by a single species of termites. During the droughts, these termites appear to have gone into a dormancy. They moved deeper underground, becoming inaccessible to our surface foraging aardvark. In summary, during typical non-drought conditions, when resources were abundant, vegetation green, and ants and termites sufficiently available to meet the aardvark's energy requirements, they were able to maintain a high and stable body temperature and remain active during the nocturnal period. Whereas during an extreme drought, when resources were scarce, vegetation brown or absent, ants and termites became inaccessible to the aardvark. Our aardvark became skinny. Unable to meet their energy requirements, they showed a very low and variable body temperature and shifted to a diurnal rhythm of activity, potentially in an attempt to reduce the metabolic costs of maintaining a stable body temperature. Many of our aardvark died. If aardvark do become extirpated from the arid region, it will have knock-on consequences for the many species that rely on aardvark burrows as thermal refuge. Maternal effects may also result in knock-on consequences, this time for the phenotype of the offspring, particularly if changes in temperature and resource availability coincide with sensitive periods of development. Mammals change their regulation of body temperature during pregnancy to protect the developing fetus. This figure shows the body temperature of two vervet monkeys over a 2-year period. The upper panel shows the body temperature of a male monkey, whereas the lower panel shows a female monkey over the same time window. We can see that the female monkey's body temperature is much more variable than that of the male. And this variability seems to be linked to the female's reproductive cycles. When pregnant, females show a progressive decline in body temperature, known as gestational hypothermia, and this has been observed in a number of species, including humans. Body temperature then reaches a minimum on the day that the female gives birth, before returning to non-reproductive levels during lactation. If we then zoom in over a 3-day window around birth, we can better see how those drops in body temperature were associated with birth. Here, the orange lines show a 3-day body temperature rhythm of 2 individuals that were observed to give birth during the diurnal period. And the blue lines show 2 individuals that presumably gave birth at night, around 9 o'clock. The black lines show the time-matched body temperature rhythm of a non-reproductive females, and the grey lines show the associated air temperatures. Irrespective of the time of day, births were characterized by this decrease in body temperature, followed by a precipitous increase in body temperature in the hours immediately post-parturition. These low maternal body temperatures may help to protect the fetus from neuronal injury during hypoxic conditions during birth. Through our remote and continuous monitoring of body temperature in free-living monkeys, we were able to accurately pinpoint the timing of the hypothermias associated with births from 16 mothers. If we then plot the timing of those births across a 24-hour day, we can see that the majority of those births occurred at night, represented by the blue dots. 2 of them occurred within an hour of sunset, when the mother was resting at her sleep site, and 2 were observed during the day, as represented by the orange dots. Compared to the non-reproductive female body temperature, birthing females reached lower minimum body temperatures and reached those levels earlier in the evening. For the nocturnal births, body temperatures dropped to 35.9 degrees centigrade on average, approximately 2 hours after sunset. Most primates, including humans, give birth during the inactive phase. In a paper recently published, we proposed that the timing of birth may have evolved to coincide with the nadir, or the trough, of maternal body temperature rhythm and cool times of day to facilitate heat loss, thereby reducing costs of thermoregulation and maximizing maternal thermoregulatory efficiency. It's not only during birth that pregnant females reduce their body temperature. Pregnant females show this progressive decline in mean, minimum, as well as maximum body temperature compared to the non-reproductive females, represented by the dotted line. Now, these data were time-matched during gestation and lactation and anchored on the day of parturition. This lower body temperature during gestation ensures a thermal gradient for heat loss from the fetus. The lower panel then shows the amplitude or change in body temperature within a day. Again, the solid line shows the reproductive females compared to time-matched non-reproductive females. We can see that particularly during gestation, the pregnant females are more tightly regulating their body temperature. But this requires energy and water. When resources were limited, pregnant monkeys struggled to maintain these narrow rhythms of body temperature during gestation, but we don't yet know the long-term consequences for the offspring. Monkeys are not the only primates likely to suffer as a consequence of heat and aridity. Pregnant women and their fetuses may be particularly vulnerable to climate change. In a recent systematic review, we highlighted reduced birth weights and an increase in stillbirths associated with elevated temperatures. These findings formed the basis of developing a heat center, which is an NIH-funded center that aims to develop innovative solutions to mitigate the health impacts of climate change in Africa. Professor Ballinsky laid the foundation for our understanding of developmental biology and the importance of concealed genetic diversity in adapting to environmental variability. Building on that knowledge will be crucial as we navigate a hotter and drier future.
think that was a very fascinating talk by Robin, and I've heard some of this before, but not all of it. And the last part about female body regulation of temperature around parturition, that was quite something special, which I didn't know either. We're going to move on the program, on to our next speaker, Professor Pavel Golden. And Pavel is based also at the Shmalhausen Institute of Zoology in Ukraine. And Pavel, you're going to talk about lifting constraints in development of cetacean skeleton. So I would hand over to you.
Thank you very much, dear colleagues. Thanks a lot, first of all, for inviting me to present here. It's my great pleasure and privilege to be here. First of all, I'm happy to present at the symposium dedicated to commemorate Boris Ballinsky. For the latest 10 years, I'm a member of the department in the Schmalhausen Institute of Zoology, where in the same department where Ballinsky has been working for
Recording in progress.
Themselves, they evolved from the terrestrial reptiles around 50 million years ago, and now they are quite small, such as the Habapopus, which can be just 1 meter long and having a weight of 30 kilograms, but at the same time there are such big whales like this blue whale, which is actually the largest, the heaviest animal, and even the heaviest living organism which has been ever lived on the earth. So it is heavier than dinosaurs or the largest trees, for example. Its weight might reach 170 ton or even more, and that's not an occasional thing. Such a diversity in body size and such a large upper limit of the body size of between toothed whales and baleen whales. Why? the answer seems to be evident, and this is among the consequences of the secondary aquatic adaptation and in the lifestyle of whales and dolphins. These animals, they returned to the aquatic lifestyle, or we might say a fish-like lifestyle, which has been abandoned by the terrestrial ancestors of modern tetrapods of more than 300 million years ago, but then they return to the sea. And what does it mean? This means numerous transformation in the skeletons. You can see it in the transformation of the skull and of the postcranial skeleton. In the vertebral column, you can see that hind limbs are missing in modern whales, and they are represented only by rudimentary pelvic bones. And also a lot of transformation can be observed in the anatomy of the forelimbs. Naming that's how the humerus looks like in a modern baleen whale, the minke whale, and this is how the forearm bones look like. This is the wrist or carpal bones. These are metacarpals, and these are phalanges of forelimb digits. Also, here you can see that there is no thumb, digit 1, and here you can find only digit 2, 3, 4, and 5. So, as you can see, all the parts of the skeleton underwent major transformation, and this is obviously because the skeleton lost the function of terrestrial locomotion. It does not have the burden of gravity. It might serve only for aquatic locomotion, and in this sense, we can talk about lifting numerous constraints in skeleton anatomy, in skeleton evolution, and it is quite interesting and intriguing to look at what was happening during the evolution. The simplest tool we applied is the investigation of anatomical networks. This means that each forelimb, each cetacean flip can be represented as a graph. And the nodes of its graph are bones, and the links between the nodes are bone articulations. In this way, we can reconstruct a flip of each cetacean taxon. Does not matter if it is a fossil cetacean or a living cetacean, if it is a baleen whale or a toothed whale. We can reconstruct its forelimb as a graph consisting of modules and links, and then we can operate with these anatomical networks in terms of the graph theory. So each graph has its own statistical parameters, which can be calculated in a mathematical way, and so we can find such characteristics as the skeleton integration, modularity, heterogeneity, transitivity, and other characteristics. And then we can draw conclusions about the integration of these parts of the skeleton, or saying further, the skeleton as a whole, why not? And what we can find that every cetacean skeleton is a complex of processes of disintegration and reintegration. Because to produce a new structure, to reintegrate, first you need to disintegrate the structure. And when we look through deep time from the terrestrial ancestors of cetaceans, then from ancient cetaceans which can be found as fossils, the whales which lived on the Earth like 30 million years ago, 40 million years ago, and then compare them to modern cetaceans, then we find that the greatest extent of disintegration has been reached around 40 million years ago. And then we are spotting the processes of reintegration which are evolving in parallel in toothed and baleen whales, and they might be quite different across the different clades of these toothed and baleen whales. So that, for example, the least integrated forelimb, which several missing elements can be found in bowhead whales. However, at the same time, we can say that bowhead whales are also the longest living whales among the modern cetaceans. And moreover, as we know now, this is the longest living mammal at present. The bowhead whales can live up to 200 years. Almost the same can be said about another cetacean with the least integrated skeleton, which is the killer whale. Here you can see the photograph of the skeleton of its forelimb, and you can see that several wrist bones are missing. And at the same time, we can say that the killer whale is a very long-living animal, and some of them can reach 100 years old. Also, what is important that all the parts of the killer whale skeleton can be characterized by so-named prolonged ontogeny. So they are very long-growing, and some of them do not stop their growth throughout the life. This can be said both about the skull and the forelimb skeleton. For example, the parts of the skull of the killer whales are characterized by isometry. So its brain case grows proportionally to the overall skull. And this in its turn means that the brain itself grows isometrically to the whole skull, which is one of the rare exceptions among mammals. As we know, in most of mammals, brain shows strictly negative allometry patterns. For killer whales, it also shows a negative allometry, but in terms of the allometry to the whole body size, but not about the skull size. And this is interesting and important, so that we can see that the disintegration in some parts of the skeleton is accompanied with a long lifespan, a large brain size, and kinds of prolonged growth, which are resulting in numerous effects of neoteny. And if we appeal to bioinformatics methods and compare sequences of several transcription factors from several families, just to say that transcription factors have been already discussed here by Vladimir Korsh and other presenters, and this is in fact our focus of our interest. So if we look, for example, the deep time evolution of couple of wrist bones among mammals and compare it to evolutionary rates and patterns of selection in several transcription factors, we find that they correlate between each other, and this means that at least several transcription factors in cetaceans underwent relaxed selection, and at the same time the increased omega ratio in their evolution. And this relaxed selection seems to be one of the major direction of evolution of regulatory genes which regulate development of cetacean skeleton. This is particularly well seen, for example, in the case of the gene HOXD13, the distalmost gene of the HOXD family, which is directly associated with the development of mangus, which means the fingers, the phalanges of fingers, and also the carpal bones. We found that there is a great variation in the polyalanine sequence of the HOXD13, which is expressed among the cetaceans. While in most of terrestrial mammals, there are only 15 and only 15 alanines in this polyalanine residue sequence, vice versa in cetaceans, so we have a variation between 12 and 18 residues. And this characteristics is also shared in several aquatic mammals, which include the walrus and sirenians. And also there is a kind of polymorphism in afrotherians, which most of them have 16 alanines instead of 15. We explain it in such a way that the polyalanine sequence is coding unordered, one of the unordered parts of the HOXD13 gene sequence, and this affects its, to say, regulatory power, and also possibly it might affect its expression during the lifetime. Indeed, we had a case study on the gene expression of several transcription factors, including HOX genes and long non-coding RNAs in the Haber-Poppers, and we found that most of them, including HOXD13, have been expressed during all the lifetime in the Haber-Poppers. One in most of mammals, including mouse and human, the expression drastically fell during the lifetime. So it should be expressed only in prenatal ontogeny or the best in early childhood, but in cetaceans, at least in the Haber-Poppers, they have been expressed in adulthood, in adult animals, and this might perfectly explain what happens with their skeleton development. It looks like an evidence for an eternal youth in these animals. Moreover, we explored in depth what happens with the evolution of the humerus bone in cetaceans, and based solely on morphology, on the geometric morphometry of the bone. And we found that while genes mostly showed relaxed negative selection in their evolution in cetaceans, the morphological characteristics showed Brownian motion. So disintegration of their evolution was an order higher than the evolution of genes, a more or less predictable effect. Well, but however, at the same time, we found another, not to say just an opposite effect in the morphological evolution of humerus. This means a strong phylogenetic signal, and the shape of the evolution of humerus shape in cetacean clades followed more or less strict parallel or divergent trajectories, which were consistent with their aquatic adaptations. So from one side, this was a Brownian motion, and from another side, this was a strongly canalized evolution toward a certain adaptation. Fortunately, this was not an effect discovered by us. It has been described by our colleagues Ryan Fellis with co-authors and called them a fly in a tube. So we can compare the evolution of such a structure like a fly which is flying in a very chaotic way, but it is flying within a kind of more or less limited space, in our case, the morphospace. So the metaphor of an insect flying within a tube in a very chaotic trajectory can be extrapolated to cetaceans and can explain the evolution of at least certain parts of their skeleton, not saying this, but possibly on the skeleton as a whole. And this is a really unusual, quite a rare situation that, to my mind, is worth further discussion and development. And I'm very happy that we got this result. That's all from me. Thank you very much for your attention.
Thank you, Professor Golden. I don't know if there are any questions online. Just for my sake, I didn't know that at least two cetaceans have indeterminate growth and in terms of the skull anyway, and then a corresponding increase in brain size. I suppose you wouldn't know there's an increase in brain activity, but there's certainly an increase in longevity. Is that related to the OX genes you were talking about?
We believe that OX genes can contribute to the regulation of longevity as they contribute to the evolution of ontogeny rate. So, and this is the issue we are studying on continuing our study right now, and we believe that these are interconnected factors.
And would they have evolved just by one event resulting in a second event? I mean, are they joined in some way, or is this one that evolved for limb development and then had a subsequent knock-on effect? So there's two selection. Is there two selection points, or is there one with a consequence? And this is what I'm trying to figure out.
That's a good question, because what we can see now, these are several processes which are going in parallel to each other, and this might be explained that possibly they could independently evolve in several planes, and these processes could involve different mechanisms. But some of them could be cascade effect, and other could be independent effects. And I believe there may be more than one solution.
Thank you. That answers my question. Are there any questions from the audience or online?
No questions online.
Thanks very much, Professor Golden. That was
Thank you very much.
Yeah, thank you. And I very enjoyed your talk about, and you certainly revealed some interesting facts about cetaceans. Thank you very much. Okay, our last speakers for today on day one are Dr. Oleksiy Bdzielia and Viktor Yepyshin, and they're going to speak about, and they're from the Institute for Evolutionary Ecology in Ukraine, and they're talking about B. Ballinsky's contribution to lepidopterology something about butterflies, and current challenging in revising Afrotropical Gelacheidae.
Neville, I will be running the presentation on behalf of Dr. Bidzila because of technical problems. So he speaks and I share the screen. Okay,
Yeah,
so we both are in action now.
Thank
Okay?
you, Alex. Yeah.
Okay. Does everybody see the presentation on screen?
Yes, we see it.
Yes.
It sounds okay. Yeah.
Okay.
Okay.
So please, Alexis, tell me next, please, and then I change.
Okay. Dear friends, dear colleagues, good afternoon. My presentation is divided in two topics. The first one, which I made in behalf of my colleague Victor Yepishin, it's Ballinsky contribution to the study of Lepidoptera of South Africa. And the second part of presentation, Micromos family Gilichidae in Afrotropical region. So the first part about Ballinsky contribution to South African Lepidoptera. His interest was restricted mainly subfamily Ficetini of Pyrroloidea superfamily. The results of his study of taxonomy of this subfamily have been published in 4 articles and 1 monograph. Altogether, described 169 species which are currently valid and 25 genera. It was made from 1987 to 1994. As you see here, it was altogether 4 articles and 1 monograph. Please, next slide. Yeah, the Ballinsky monograph, it was his final works devoted to Lepidoptera of South Africa. It names a study of African Pizzettini in the Transvaal Museum. It's the most important contribution to taxonomy of South African Pizzettini. In this monograph, 135 species and 19 genera were described. As you see, the monograph is perfectly illustrated by hand drawings and monochrome photographs of adults, and also their heads, wing venation, and genitalia of both sexes that allow to recognize most of species which are known to Ballinsky from South Africa. Please, next slides. Besides Pizzettini, Boris Ballinsky was interested in another group of Lepidoptera of the Afrotropical region. For example, the National Museum of Natural History in Kiev hosts a Lepidoptera collection by Ballinsky, collected by Ballinsky in South Africa from 1950 to 1991. This collection was donated to museum when Ballinsky visited Ukraine in 1994. Please, next slide. Is it?
We have sound problems.
Please unmute yourself.
I think he might have muted himself. Alex, can you hear me?
Yeah, I can hear, but Alexis is in Belgium now, so we are in different countries. And yeah, I just can make a phone call, just a second.
Okay, thank you.
Sorry, some technical problems. So this collection consists information about 35 families of butterflies and organized in the 40 wooden boxes and more than 500 microscope slides of genitalia and wind venation. As you see, most specimens collected by Ballinsky are in good conditions, well prepared, labeled, and can be used for further study of Afrotropical Lepidoptera, and they are also partially identified. It should be also mentioned that one new genus, Ballinskya, and one new species, Nonficita ballinsky, were named in the honor of his in recognition of Ballinsky's contribution to study of South African Pizzettini. It was the first part of my topic, and please the next slide. It's some results of my study of Micromos family Gillichiidae. Gillichiidae is one of the most species-rich family, not only Microlepidoptera, but other Lepidoptera in the whole. Worldwide, more than 4,000 species are known, of which about 1,000 in Europe, in the Arctic region, and just 567 species were known in the tropical region by 2002. Please, next slide. Among Gillichiidae, there are some species which are known as economically important pests, which have been introduced to many countries of the world, and among them are most dangerous is Tuta absoluta, Hesperia lycopersicella, Citotroga cerealela, and Pictinophora gossypiella. Please, next slide. Here are some words about history of study of this family in Afrotropical region. First publication, first description of Gillichiidae were made by Christoph Ziller and Lord Walsingham at the end of the 19th century. After that, Edward Meyrick described more than 300 species of Gillichiidae from South Africa, Namibia, Mozambique, Zimbabwe, and Central Africa. After that, Janse compiled all data of South African Gillichiidae, revised, redescribed, and illustrated nearly all previously established taxa, clarified their taxonomic and generic segment, established many new genera, and described many additional new species in his book, Most of South Africa. After that, additional contributions were made by several authors, first of all by Vary, Powolny, Zattler, and all these data were summarized in two catalogs of Lepidoptera of South Africa. After that, from 2002 to our days, several faunistic and taxonomic papers have been published. Totally, 567 species of Gillichiidae have been recorded from South Africa by 2002. Please, next slide. What tools are available for identification of Afrotropical Gillichiidae? First of all, it's a monograph by Janse, Most of South Africa. It was published in 2 volumes and 7 parts from 1949 to 1963. This next slide. This monographs contains information about more than 500 species of Gillichiidae from Namibia, Zimbabwe, Mozambique, and South Africa. You can see here there are monochrome photographs of adults and genitalia structure, and also drawings of wing venation, genitalia, both males and females, and diagnostically important parts of the heads and some other details. Please, next slide. The next sources of our knowledge is, of course, scientific papers and catalogs devoted to different groups. It's taxonomic revision, it's catalogs of collection which contains information about type specimens, it's faunistic papers with separate description and faunistical lists. Next slide, please. It should be noted that in last years, the special series of books, Esperiana Memoir, was establishing to publish the most important result of study of biodiversity and taxonomy of Afrotropical Lepidoptera. Regarding Delichiidae, in these books, three papers have been published, one of them devoted to Lepidoptera of the Brandberg Massif in Namibia, and also a Santasana game farm in Eastern Cape of South Africa Republic in Graaff-Reinet district. Please, next slide. And very important things, it's electronic database AfroMOS. It's the most complete, regularly updated online database of Afrotropical Lepidoptera. You can find here all necessary information about current taxonomic states of species, about original publication, pagination. Please, next slide. For example, you can see author, type, locality, geographical coordinates, and many other information. And next slide. And of course about distribution with references to publication. This database is regularly updated and considered as the most complete database about all Lepidoptera order in the Afrotropical region. It's curated by Jurate and Billy de Prins from Belgium. Please, next slide. And some words about collection, because collection remains among the most important sources of our knowledge of Lepidoptera diversity and Delichiidae in South Africa. Among the most important depositories of Delichiidae are the National Museum of South Africa in Pretoria, it better known as Transvaal Museum of South Africa. This next slide. This collection is famous due to a huge number of type specimens described by Edward Meyrick and Antonio Janse. Next slide. And besides this, collection hosts a very rich material collected by its former curators, Laszlo Bari, Georg van Cornelius Riestra, next slide, and of course Martin Kruger.
Alex is muted again.
Suddenly, the technical problems of he really joins us.
Okay, maybe you can unmute then.
Yeah, it's not he's muted, he lost connection, so he reconnects. Just a second.
Oh, okay, thanks.
Yeah.
Are you hearing me?
Yes, I'm here, and he's trying to reload. Just a couple of seconds. I will try to call again. Maybe I will put on the dynamic my mobile, and you will hear him from my mobile, and I run presentation. Is it a good solution? What do you think?
Alex, were you asking whether you can do feed from his cell phone, mobile to your mobile?
Yeah, this is what I will do.
If you can hear me, yeah, that's a good idea.
Just a second. We will continue now. Sorry, we continue from my cell phone.
So in this slide you see Martin, the former curator in the Museum of National History, the outstanding collector of all groups of Lepidoptera, including microlepidoptera. Next slide. The collection is organized in classic style, wood or plastic drawers for specimens, with type specimens keeping in separate isolated room. And as you see here, there is unique way of keeping the genitalia slides. This next slide is British Museum of Natural History in London, holds one of the richest collection of tropical Orchidae. Besides types of Myrick and Walsingham, the collection holds rich material collected in various regions of Africa. For example, 1,000 specimens collected by Urate prince and Billy prince from Congo, Cameroon, Kenya, South Africa are kept in this museum. David Agassiz, Western Supermare, United Kingdom, collected the Glycidae in Kenya, Tanzania, Uganda since 1998. His collection is widely used as a platform for taxonomic revisions of some groups of subtropical Glycidae. This collection will be transferred to PMNH soon. Next slide. Also should be mentioned is the National History Museum of the University of Oslo, which also hosts rich collection of Glycidae by Leif Arvik from Uganda, Kenya, and Tanzania. You can see also Wolfgang May, the former curator of Lepidoptera and Trichoptera in Berlin, and the founder of one of the most significant collection of the tropical Glycidae collected by him in various countries of tropical region, mainly in Namibia and South Africa. Next slide. Other important depositories of tropical Glycidae are the Royal Museum for Central Africa in Tervuren, National Museum of Natural History in Paris, and South African Museum in Cape Town. Please, next slide. And now some my suggestion and some problems with species identity I faced during my work with tropical Glycidae. The first case is raise a common things. The species is described from single specimens, which holotype is not traced in the collection. The type can be either destroyed by pests or just missed. what decision for this case? The most typical is looking for type in the collection in hope that type specimens will be found. If not, we are looking for additional externally similar specimens, preferably from type locality, as they confirmed by molecular data. And in some exceptional case, neotype designation is necessary. Next slide. The second common problem is the species is described from single specimens that lack the abdomen and no additional material is available. Decision the same, looking for additional externally similar species, their identity confirmed by molecular data. And the next problem, when the only available holotype is a female, these characters are not informative enough to judge about correct generic segment or species. Originally, such species usually placed in so-called collective genus. For example, Triphusa ampichroma was described from single female in collective genus Triphusa. Currently, this genus is restricted to neotropics, whereas afrotropical species originally described here were transferred to other genera based on male genitalia characters. If they have the only female, we can say nothing about the generic assignment of the species until the female will be found. In this case, the decision is the same. We are looking for additional externally similar specimens of opposite sex, desirably from type locality, and identification, of course, must be confirmed by DNA study. Next slide is unusual variability in external and genitalia characters of some taxa. For example, African specimens of Citatotrops acasta, as you see, show considerable variation in the male genitalia. It is unclear if they represent one variable species or replace triptic diversity. The situation is complicated that there are two additional species described from Eastern Asia, Zengi and Celtops acasta. They are most likely conspecific to each other, but their conspecificity to Citatotrops acasta and or some of junior synonyms of this taxa should be proved. And next slide. Some my suggestion about current knowledge about the family and genera groups of afrotropical Glycidae. At this slide you see the members of subfamily Apatetrini. About 13 genera, 10 of them are monotypic, others comprise few species only. The South African genera Filisignella, Curvisignella, Ichneocraspedus are known from females only, hence their current taxonomic position is rather provisional. Most of undescribed species belong to Apatetris, whereas genera Anomaloga and Pluricista are most likely congeneric and should be revised with the description of several new species. Next slide. Some species of this subfamily are later distinguished externally in the genitalia, having, for example, deeply excavated hindwing or strongly asymmetrical genitalia, and generic assignment of some species like here are rather unclear. The tribe Aristoteliini, a very species-rich tribe which comprises about 14 genera with more than 70 described species. For example, the genus Aphanostola comprised a small moss 5-10 millimeters in wingspan, which larva feed on acacia. Before my study, only 2 species were known, but as a result of revision, additional 18 species from afrotropical region have been described. Next slide. It's subfamily Geotrichini with 4 genera and about 32 species. Polygymno is very diverse in afrotropical. Janssen in his book listed 24 species for South Africa. However, a number of undescribed species are known from Eastern and Central Africa and waiting for their description. Next slide. So family Gillichidae comprises about 15 genera with more than 120 species in South Africa only. Many species remain undescribed within Triceratops poison. Very similar, causing problems in separation of externally similar species. The revision of the genus seems hardly possible without DNA study. The next slide, Nitini. Rare species reached is represented by 12 genera and about 35 species in South Africa. Several new genera must be established. But on the other hand, the group of genera related to Teleiodes must be revised and probably synonymized with each other. The most important and remote problem is the genus Telfusa. 9 species described in this genus are known from females only, but their generic segment have declared flight when the males are discovered. All species described are divided between 3 genera. It's Strama, Vatiman, and Arze. A large number of undescribed species are known from the 2 genera, and Arze has been studied quite satisfactorily in South Africa, whereas many new species are known in other regions. Next slide. In this genus there is also some problems because genus comprises at least 2 groups of species. Because the male the type species belongs to the group by presence of modified scales of the deep vulva and absence of natus in the male genitalia. The developed hook-shaped natus vulva not bearing is broader than the left vulva. This is sufficient to justify the establishing a separate genus. A very diverse species range groups. The number of undescribed species in the is difficult to estimate, especially in Eastern and Central Africa. In general, Ticomeridini from South Africa are studied better than those from the rest of the Afrotropical regions. At last, half are we from the next slide. And some words about my participation in Santa Santa project. This University of Santa Santa Game Farm in the Karapagna district in Eastern Cape of South Africa. The material was collected, please next slide, from 2007 to 2013. Next slide, 2012. Next slide. The material was collected by light trapping and daytime collecting. Next, probably the material was sorted and prepared in the field next morning after being collected. Most of the specimens were pinned, and 1-2 specimens, almost interesting species, have been spread in the field to get well-preserved samples for publication. It should be the next slide about results. The project resulted in publishing series of papers devoted to biodiversity and taxonomy of 22 families of Lepidoptera from Santa Sana locality. Regarding Delichidae, 124 species were recorded, of which one genus species were established. The second part of this monograph is preparing now and hopefully will publish it in coming years. And slide some conclusions. Since 2002, the number of Delichidae in South Africa, Namibia, Mozambique and Zimbabwe ponds increased from 567 to 622 species. 7 genera have been revised, 7 genera have been described, 134 species from 32 genera have been described from South and Eastern Africa. 26 new genera combinations have been proposed and 10 new synonyms have been established. Also, 87 new host plants have been recorded for larvae of Delichidae. It's mainly in South Africa, in cooperation with German Staude. Despite considerable progress in study of Delichidae in Eastern Africa, in the last 15 years, the family is still studied very incomplete. Many genera are in need of revisions and that number of species are still undescribed. In general, South Africa is studied most satisfactorily in comparison with the rest of the tropical region. The study of collection material shows that at least every third species from Eastern Africa is undescribed. And another conclusions, our knowledge of Delichidae from Central Africa is much more fragmented. Separate description of several new taxa or record of pairs of crops. And the real level of species diversity in Central Africa can hardly be estimated. And the next slide, this is some of my plans for next 2 years. Is a series of museum to study additional material, to revise some genera, and updating the information in Afromos database. And final aim is preparing illustrative guide for identification of the most common genera and species of Delichidae and in South Africa, Republic and Namibia, based on external characters, inclination, morphology of head, terminal segments of abdomens. It will be made in cooperation with Wolfram May. Thank you for your attention and please sorry for technical problems.
Do people hear us? Neville, I don't hear you. Ah, yes, okay. Now we don't hear you, Neville.
Oh. We can hear you. I don't know how we tell you that.
Yeah. Okay.
Can you hear me?
Now. Yeah, now I hear you and Alexei hears as well.
Okay, I don't know if we can take questions because it's going to be very difficult to route it through you. But if there are 1 or 2 questions from anyone, I'll take it now and then we can wrap up the day. Anyone? I don't see anything and I can't see online either. But I do wish Alexei all the luck, and I see he's coming out to South Africa in terms of some of his research, and if he does come out to Ditsong, he should please come over and visit us at the university in Johannesburg. But I think we'll wrap it up there, and good luck with everything, Alexei. It's a very interesting study, it's quite a comprehensive taxonomic revision, and it's not easy from what I see, but all the best.
Yeah.
Thank you.
Yeah, and I would like to add that through the presentation of Alexei, we can see how difficult work of Boris Ballinsky was, because he also was through all this process of preparation of genitalia, finding tiny characters, and also he was combining it with his embryological and teaching and administration work. So, like, we can see how difficult it is to be lepidopterologist, and in the case of Ballinsky, it's especially how difficult it was to combine it with all other activities. Okay.
I agree, Alex. He was an incredible scientist and just a general human being. So thank you for raising that. With that, we end today's session. We'll see everyone in the morning, and we have a new series of speakers. So thanks everyone for joining in, and all the best. Thank you.